डिस्प्ले ऑन कर देना।
देखो तो ये
ठीक है। of the organizing committee, I welcome our distinguished guests, Professor Akhilesh Verma, Dr. Bhavesh Dev, Professor Ram Singh, Dr. Gaurav Krishnan, Dean of Faculty of Sciences, Professor Sanjeev Thakur, Head of Chemistry Department, Professor Ranjana Sarkar, faculty members, and students, to the first meeting of Organic Chemistry series, which is on the theme of harnessing the catalysis for green organic synthesis. I now request guests and dignitaries to come on the dais and take their seats. Now I request Dr. Sushmita, the convener of this program, to present a bouquet to our guest, Professor Akhilesh Verma. Thank you, Dr. Sushmita. Thank you, sir. Now I request Dr. Uma, the co-convener of this program, to present a bouquet to Dr. Bhavesh Dev. Thank you, sir. Thank you, Dr. Uma. Now I request Dr. Swapnil to come forward and present a bouquet to our Dean, Faculty of Sciences, Professor Sanjeev Thakur. Now I request Dr. Jugal to present a bouquet to Head of the Department, Professor Ranjana Sarkar. Now I request Smriti and Sheetal to come forward and present a flower pot to Dr. Uma and Dr. Sushmita, who are the organizers of today's events. And without them, the commencement of series was not really possible. They are the backbone of this program. Now I request the dignitaries to come forward and light the lamp. As we all know, green chemistry is the tool for realizing the sustainable and economical chemical transformations. By minimizing hazardous materials, reducing waste, and promoting energy-efficient processes, green chemistry contributes to the healthier ecosystem. So, there is a growing demand for finding new ways to improve the conventional methods by performing chemical reactions that are kinder to the environment. And researchers like us have the great responsibilities here. 12 principles of green chemistry are the guiding light for us to start our research directions. Among them, catalysis is one hero which ensures more of the starting materials end up in the desired products. Therefore, for today's session, we have invited eminent researchers from the prestigious institutions who have been contributing to this field and are here to enlighten us with recent advancement and future directions in this field. The series has been started with the aim to give exposure to our students and researchers about the current focus of the research. This is a step forward toward our contribution for the developed nation by 2047 and our contribution to the campaign recently initiated by our Honorable PM, Shri Narendra Modi. And we hope to bring more such meetings in the future for our students. I request everyone to kindly be engaged and witness the wonderful research outcomes. Now I request Dean of Faculty of Sciences, Professor Sanjeev Thakur, to come forward and say a few words about the program.
Good morning, everybody. Dignitaries sitting on the dais, Professor Akhilesh Verma from University of Delhi, Dr. Bhavesh Dave, he is from pharmaceutical industry. Two more speakers are about to come, they are yet to arrive. Professor Ram Singh from DTU and Dr. Gaurav Krishnan is the senior principal scientist from CSIR. So dignitaries on the dais, dignitaries off the dais, all my dear faculty of physics and chemistry, I guess some of the maths people are also sitting over here, and my dear students of masters, PhD students, and some of the BTech students, very relevant topic, that's why I bothered to call you people. Some of them are sitting here, BTech students as well, because they are also studying the green chemistry. And this seminar is probably about this significance of organic chemistry, sustainable energy, green catalysis, and drug discovery, and engineering-related inventions. The speakers, the eminent speakers, are going to throw the lights on these topics. And I'm extremely happy that they have come over here. They have spared their valuable time, and they have come over here to give some exposure to all of us, and especially the students who belong to BTech and masters and PhD students. So it's an honor to share the dais with dignitaries like Akhilesh Verma and Dr. Dave. First and foremost, welcome you all in the inaugural function of this seminar. And I will also take this opportunity to congratulate the organizers, especially the head of the department, Professor Sarkar, the coordinators, Dr. Uma, Dr. Sushmita, Professor Purnima Jain, all the young faculty members of chemistry department, Dr. Jugal, Dr. Sonavane, Dr. Sunita, Dr. Shikha, Dr. Nancy. I think I missed your name, but you are the star campaigner. So And I'll take this opportunity to congratulate all of you that you people have uh, organized this in such a short span of time. And this has been possible because you pursued the matter, and within 4 or 5 days, this has been organized. So I'm glad that you could do it, and my gratitude to the Honorable Vice Chancellor that he took no time in approving the things. And we took the file to him, and straight away he said, yes, I'm going to encourage my students. And that way, it's actually an exposure for you people that all the topics are so burning, so hot, and so relevant in the present scenario that you will certainly be enlightened. And that way, our Honorable VC and the account section, the administration, all of them have given the full support to make this happen. Many more bigger things are going to happen in a few months. And these youngsters are planning to go ahead with those programs. So this seminar has actually fructified in a very short span of time. And all the financial implications have also been taken care within no time due to the support of everybody here who are important. And with these eminent speakers on the dais, you all will get acquainted with the burning and relevant topics like what I said, sustainable chemistry, drug discovery, green catalysis, energy-related inventions, and a fruitful interaction is going to happen, I'm sure about it. So this seminar is going to be fruitful and productive for all of you. That's what I guess. And my best wishes to all of you. Thank you.
Thank you, sir, for addressing the audience and for your encouraging words. Now I invite Professor Anjana Sarkar, head of the department, to say a few words about the program.
Very good morning to all of you. I welcome our distinguished guests, Professor Akhilesh Verma and Dr. Bhavish Dave. Professor Ram Singh and Dr. Gaurav Krishnan will be coming later on. Then Dean of Faculty Sciences, Professor Sanjeev Thakur, all the faculties, members, students, and on the first meeting of the Organic Chemistry series. We extend our sincere gratitude to the esteemed speakers for accepting our invitation to join us and present their research findings. It brings us immense joy to provide this valuable opportunity to our students to gain insight into the current focus of research. We have started this lecture series for our students, aiming to enhance their educational experience and prepare them for future endeavors. Such lectures also give them exposure to diverse areas of research. Such platforms also give researchers an opportunity to stay updated with the latest developments in their field. Thought-provoking scientific discussions to improve their own research, inspiring them to integrate ideas from other fields into their own research. I, as the head of the department, would love to witness more such lectures in the future and would be ready to help in any regard. I also thank Dr. Uma Narang and Dr. Shushmita for their efforts in bringing this series to life in our department. I request everyone here to take the best out of this opportunity. And obviously, the last but not the least, I am grateful to our Vice Chancellor, Sir, who has been encouraging us, and he also has requested that we carry forward such endeavors in future. Thank you.
Thank you so much, ma'am, for sharing the agenda of this program. Now we will all assemble outside for high tea, and we will be back in 25 minutes for the commencement of the program. Thank you, everyone. Just outside we have high tea. This side, this side. Students, please settle faster. So welcome back to the event, everyone. We have good weather, nice weather, and we had good tea. So we are all now geared up to listen to the presentations we have. So now I request Professor Anjana Sarkar to chair the next session and introduce our speakers and take the session forward. Thank you, ma'am.
So, now Professor Akhilesh Kumar Verma, he's from University of Delhi. Dr. Akhilesh Verma received his PhD from the Department of Chemistry, University of Delhi in India in 2000. He carried his postdoctoral studies in the University of Florida, USA, with Professor A.R. Catrizzi from January 2000 to December 2000, and with Professor R.C. LaRocque in June 2007 to August 2008 at Iowa State University, USA. He is also a recipient of Boy Scout Fellowship from DST India. Presently, he is serving as professor at the Department of Chemistry, University of Delhi. His current area of research focus are on heterocyclic synthesis, hydroamination of alkynes, then tandem reactions, iodocyclization, CH activation, fluorination chemistry, and development of new synthetic methods. Currently, he has more than 140 publications in leading organic chemistry journals. Professor Akhilesh Verma.
So, thank you. Thank you, Professor Anjana, for your kind introduction. And before starting my lecture, I would like to thank organizers of this event, Shushmita, Uma, and also I would like to thank the Vice Chancellor and Dean of Faculty of Sciences, because without their support, it's very difficult to organize any event. So my sincere thanks to Honorable Vice Chancellor and Dean Faculty of Sciences for supporting the organizing committee. And when I look this title, Harnessing Catalysis for Green Organic Synthesis. You know, the word catalysis is there, but the catalysis is not only for the chemistry. The word catalysis is also for the young students. For the growth of the country, the catalyzing the young minds is very, very important. Because the young generation is the future of our country. This is what is really needed at this current time, because I can see there is a decline in the field of basic sciences. The students, turning the students in the field of basic sciences in the last 10 years has been declined. So question is to why this decline is there. So after the masters, and always the students think about what they have to do. They always think the faculty position and position of scientist. Most of the time, people will always think the future in government sector. But let me tell you one thing. There is a lot of scope in pharma industry. India is going to the leader in coming years in the field of pharma. And as our Honorable Prime Minister has already mentioned, after this, our clashes with the China. So we have to make everything make in India. And that Make in India initiative, since then, a lot of pharma industries has opened the new market. And they started several companies. The chemists, they themselves have started. And when you look the future in pharma industry, it's open. But it's always like in the beginning, you won't get handsome salary. But after some time, and it is always depend upon your performance. How you perform in industry. When you perform well, you can go up to 50, 60, 70, 80 lakhs per annum. So this what I think most of you people don't know about this, your future in the pharma. Beside this academics, the future is there in the pharma. So always you can try, but the only thing is that you need to work hard. I know it is tough time for all of you because number of competent has increased. So you have to prove yourself, whether it is academics or it is pharma. Competition is tough. You have to find your position yourself. It is all depend upon you, how much you can work. How much you can work hard. Right? So this is my message to all of you, young people. So you can see today's my title. The construction of small organic molecules. In the chemistry, you might have seen organic chemistry making a bond, 1 bond, 2 bond, 3 bond, like that. But at present, most of the chemists, they are turning up towards the sustainability, right? And green chemistry. The question is, from where the green chemistry term has come and why all this has been started. So there is always some reasons. So this is what you can see here, the sustainability, right? The sustainability is not, there are two terms. Now you can see, and this is, we have to protect our environment and globally also. So method which is developed that should sustain for a longer time. It should not be only for a short time, or if you have any very expensive or very tedious, that is not sustainable. It should be very simple and useful and the cost effective. So there are some major incidents from the past, and because of that, this green chemistry or sustainable chemistry term has come. And this is, you can see, 22nd June 1969. the Cleveland, Ohio. It is strange but true. The river in Ohio becomes so polluted, it caught fire. This is what you can see here, the river before this accident. So, whenever any accident happens in the US, it always gets attention. But not in India. Anything which happens in US and Europe, it always comes in limelight. So this is the picture you can see here. And close to this river, there are some pharma industries, factories, and the pollutant was coming to this river, and the river got caught fire because, the solvents. In organic chemistry, you use always the solvent. So when all these effluent was going to this river, and there was cleaning was going on in this, and these people were doing this cleaning of this river through the boat, and because of some spark, the whole river got caught fire, and these people burned alive on that river. And you can see there was one bridge on that river that was completely destroyed. And this is the picture you can see after that accident. So this accident was in the limelight in all over the world. Then another accident was in Italy. Now you can see this reaction, right? I think all of you can see this reaction. This is a substitution reaction. So this substitution reaction you can see, and most of the accident happened in this pesticide company, right? So if you look this one here, it is this is tetrachlorobenzene, and this is a solvent, ethylene glycol, Na, which is a base. So with the replacement of this one, it is all depend upon how much base you are using. So always some side product is always there. So this is 2,4,5 trichlorophenol. But beside that, this dimerizes and makes this TCDD, 2,3,7,8 tetrachlorodibenzodioxane. It's called dioxin accident. And this accident was happened in Milan, Italy in 1976. And now look here, what happens? There was 600 kg of TCDD. So this is called dioxin. Cloud spread 50 meters to the atmosphere. And in less than 2 hours after the accident, the chemical becomes settled on the soil on this nearby area, 6 kilometers. Right, so this was the second accident. And this accident release of chemical including dioxin. So mainly this was the chemical which was released from there, right? Because of this accident. And now, coming to the most, worst disaster. I think all of you know this, right? And I am witness for this accident because I was staying just close to that, and my friends, because I was in Jhansi, and my friends, they were living in Bhopal. So, December 3rd, 1984, 2 a.m. midnight. And it was so cold. And whenever you have the fog and cold, the moisture is there, right? And that also was one of the cause for that spreading of that. So, and this is a union carbide, all of you know, right? And why this accident was happened? Because the safety reasons. Because of the safety reasons. And that is why whenever you are doing experiment in your lab, the safety is more important. The most important concern in the lab is the safety. One minor mistake can destroy whole lab. And this was deliberately, because they don't want to spend more money on such kind of tanks, because they knew this thing. There was some problem is there, right? And sometimes we are also responsible, our people who allowed this thing, right? And you can say this. You might have seen the movie, some of you, right? And actually this become a whole, is like gas chamber. Methyl isocyanate, a small molecule. It's the most reactive, right? Highly reactive molecule. And once you have water, right? Highly reactive, then, and it was the complete tank where methyl isocyanate, you can see here, 27 tons. And there were tanks in that methyl isocyanate was there. And because of that leakage, because the tank was not proper, that wall, it has came out, reacted with water. Huge pressure was generated, it was blast. Right? All gases reacted together. The most deadly was HCN. When you look this whole reactions, the HCN was generated, right? That come out, HCN, carbon monoxide. There are several gases, not only this. Pressure was so huge and all reaction took places. Temperatures was very high. So ionic and free radical, all the reaction took places. And this is how this HCN was generated. Right? And you can see here how many people, more than 27,000. But you will get different figure in different places. Right? So, and this was in Old Bhopal, near to railway station, Union Carbide. That is called Old Bhopal. Right? So, cause of the Bhopal gas tragedy, none of the safety system at plant were functional. So, Union Carbide own document proves the company designed the plant with unproven and untested technology and cut corners on the safety and maintenance in order to save the money. This is the main reason. Right? And the chemical that Carbide abandoned in the around their Bhopal factory have contaminated the drinking water. It goes through the soil and is spread all over. So drinking water was contaminated. Now still, the study is going on. After some years, they do the survey, and you can see here, sample of the groundwater from wells around the site showed high level of toxic chemical including chloroform and CCl4 and several other halogenated salt. It was observed. It is simple, but just you take the sample, analyze through the GC, and you will get the peak. You can say this is whatever is there. So now you have a sophisticated instrument. By using that, you can easily analyze what kind of, the impurities and what chemicals is present, right? And this is, you can see here, the testing report published 2002 revealed poisons such as 1,3,5-trichlorobenzene, dichloromethane, chloroform, lead and mercury in the breast milk of nursing womb. So what you can expect now if you have such kind of thing is still in that area. So you can see the after effects of this tragedy, right? Now you can see this is after that, and it's still. So you can see one accident, and so after all this, so many accidents. The Sustainable and Green Chemistry, the Pollution Prevention Act of 1990, right? So what is this? This was the US environmental law stating that the first choice for preventing pollution is to design industrial processes that do not lead to waste production. And that was mainly for the industries, chemical industries and others, where the waste is going, right? So what was this main outcome of this? One needs to design in such a way so that waste is minimized or there is no waste, right? Making chemical products that do not harm either our health or the environment, using industrial processes that reduce or eliminate hazardous chemicals. And now many countries have signed these international treaties to reduce pollution level. So in May 2001, that's called Stockholm Treaty, right? So the global treaty on persistent pollutant. I think all of you know, in Rishikesh, we used to have the DDT plant, right? Now there's no DDT plant. Because of this, persistent pollutant means those can continue for a longer time, right? So POP are toxic chemicals that adversely affect human health and environment around the world. So under this treaty, known as Stockholm Convention, countries agreed to reduce or eliminate the production and use and release of 12 POPs. And all of them you can see here, they all are insecticide or fungicide. Look here, these, Eldrin, Chloride, DDT, Dieldrin, Endrin. So all these are halogenated. All these are halogenated. And DDT also is halogenated. And because the effluent was going into river Ganga, right? And that's why it has been now banned. One of the 12 of this, right? So if you look here, just Eldrin, you can see here how many chlorine molecule is there, right? Chloro. And we have a negative as well as we also have the positive points, right? We have all the drugs, medicines, all. And nowadays, everybody, is whenever anything happens, you take the medicine and next day you are fit, right? So you can look here, the benefits of chemical industry, antibiotics, medicine, fertilizers, plastic, nylon, gasoline, water purification, all, right? So we need to have chemicals also, but in sustainable way, right? So these are key steps. Look here, Pollution Prevention Act 1990. And this is called GC, begins in 1991, right? And 96 Presidential Green Chemistry Challenge Award. And 1997 Green Chemistry Engineering Conference was organized. In 1999, the Journal Green Chemistry started. The Journal Green Chemistry has very high impact, right? More than 9. And Chemical and Engineering News was started. In 2001, Journal of Chemical Education was started just after this. So that everybody need to make their focus on sustainable catalysis or sustainable way for making organic molecules. Now, efforts towards sustainable chemical synthesis. Those who are in organic chemistry, now I'm coming to slowly into the organic chemistry. The base, now you know why this sustainable term has come and green chemistry term has come. So These are the reaction, like cascade reaction, domino reaction, tandem reaction, multi-component reaction. All of them, these are term comes after such kind of these incident, though it was long back before that also, but got more attention after these accidents, right? So in these, you can have a multiple formation of bond, or if you have a 3 compound take together, so 1 bond formation, 2nd, 3rd, and 4th, like that, and you will get the product. Let me tell you one thing. When we are doing any reaction, sometimes you see the first bond is formed, right? After that, next, then next, then next. All this depend upon the energy. You get first bond, right? That becomes strong. Then that will not cleave. Then the molecule which is having another reactive side with a close proximity will take the bond formation, and this is how the small molecules can be generated. So look here, the transition metal-free chemical synthesis. Now, most of the organic chemists, they are trying to design the method in which the metal is avoided, transition metal. And why this transition metal is avoided? High toxicity of heavy metals compound, and some side effects, and low abundance. So there are several reasons, right? And because if you look, there are several reactions where gold is used, rhodium is used, platinum is used. They are highly expensive, are not sustainable, right? And designing of organic reaction for particular molecule, you need to have a smart thinking, basic chemistry. If your basic fundamentals are clear, whether it is chemistry, physics, and biology, or anything, when you are able to think, then you can easily design. So now you can see what we are doing in our lab. We are playing with the 2 building blocks majorly. This is orthoalkynyl aldehyde, and this is orthohaloaryl alkyne. And all of you know, in 12th and 11th you have studied alkynes, right? And you can do the addition reactions. Bromine, water, right? So idea is from there only. When you look here, what I have designed is not very complicated. It's a simple chemistry. Only thing is that you have to design in such a way so that you can construct a useful molecule, right? And these starting substrate can be constructed from respective halide and alkyne. When you have halide, you can do the Sonagasha coupling reaction, right? And you can construct these kind of building blocks very easily, right? And then I am looking the application in our lab for making variety of useful molecules. Now, first you can see here, iodine-mediated selective oxidative esterification and electrophilic cyclization. This method, when you have any aromatic aldehyde, you can directly convert into ester without formation of acid. And this we have introduced as a green experiment in master's practical in DU. And you can see here, if you look this molecule, you have alkyne, aldehyde, and alcohol. 3 groups are present, right? And when you treat this with iodine and alcohol, you can convert selectively only this aldehyde functional group into ester without affecting this alkyne and alcohol group. So this is called chemoselectivity, right? So two groups are unaffected, only one is transformed into ester, right? By manipulating the reaction condition, you can also convert another, this two, as well as this aldehyde, as well as alcohol into the ester without affecting the alkyne. So you know when you're using aldehyde, alkyne is there, iodine is there, right? So reaction can take place on alkyne also, but the preferred reaction is on aldehyde. And alcohol can easily oxidize into aldehyde, and then it can turn into an ester, right? So you can see here, these we have published two papers, ChemCom and JOC. And if this is aromatic aldehyde, you can convert into ester. Orthoalkyne having formyl group can be okay. So now you can see here, and this can be further converted into this iodocyclase compound and these compound. So, and all these are useful molecule in biological point of view. I am not showing here the relevance with the biology, I am not showing the example, but all of these are useful, right? Whenever any organic chemist try to make the molecules in the introduction part, they always mention the relevance with the molecules with the biology or the importance. Now look here. If you look here very carefully, this reaction is aldehyde alkyne, orthoalkynyl aldehyde, iodine 2.5 equivalent. Whenever you do any reaction, how many equivalent reagents you are taking and which solvent you are taking makes a big difference. And always, when you do any reaction, please pay attention always on the side product. Side products sometimes are very, very useful and will give you the new chemistry. And how this reaction goes? And you know, the iodine forms iodinium intermediate, right? And this aldehyde, oxygen is a lone pair, right? When they are in the close proximity, minimum bond difference. Now look here. This oxygen has a lone pair. This will attack regioselective on this carbon, and you will get this Y intermediate. After using this lone pair, oxygen will get a positive charge. Once you have positive charge, now you have the bond between oxygen and carbon, right? And which is more electronegative is oxygen. So now, after that, this carbon become a more reactive, and alcohol, or you can say any nucleophile can attack here, and this is how you can generate this molecule, right? And this condition and another condition, when you look here, another condition. Here, dichloromethane was a solvent, and in this, only the thing is that iodine and alcohol is used as a solvent. The only difference is the solvent. Now, in this case, how this reaction goes? It first forms hemiacetal and then hypoiodide, right? So the OH first attack here, right? And then H is replaced by iodine, so you get this one. And after that, there is release of H and I, and you get directly ester. And here, the thing is that both electrophile and solvent are alcohol. Here, it is dichloromethane, where iodine solubility is more, so you will get first iodinium intermediate. And once you have this, right? And this further you can treat with the iodine in dichloromethane, and this will attack here, and then you can have this useful molecule. And also, the incorporation of this iodo handle. And this is one very common reaction of coupling reactions. When you have any halide, you can do variety of coupling reaction and you can introduce a wide number of functional group. This is what is in several of industries, people are making large number of molecule for biological screening by using iodo handle. So all such kind of molecule could be constructed by using this chemistry. Here you can have any kind of other. You can have this benzene ring. You can have this, quinoline, pyridine, and benzothiophene. So all different kind of molecule could be constructed, and R can be variated, and OR could be a different group, and iodine further can be functionalized. So whenever you do any synthesis, important is characterization. Proton NMR, carbon NMR, HRMS, and finally, because sometimes you may get a regioisomers, two possibilities are there. One can have a 5-membered and 6-member ring, right? And both will have a same number of carbon, hydrogen, proton. So finally, what you can do single crystal XRD, right? And that will give you the right picture of what you got it, fine. So this clearly indicates the product we have formed is the correct one. Always when you do any reaction, there are some limitations, right? When we tried reaction with tertiary butanol, bulky alcohol, so all basic chemistry, ethanol, propanol, and butanol, all they have a different reactivity towards, right? Because tertiary butanol is a bulky, so it did not give this desired structure. Rather than what we got is this iodocyclized product. And the reason is very simple. We did not, with this, after this, because of the bulky nature, the hemiacetal after that, the iodine reaction this is not forming. So rather than after this, immediately after getting this attacks, this forms an iodinium intermediate and attacks takes places. This is how you will get this iodocyclase product rather than getting this ester. Limitation with phenol. So there is a reactivity difference between alcohol and phenol. And phenol, all of you know, is resonance stabilized and 4 position is highly reactive, right? So 4 position is highly reactive. So what we got here, rather than this, the reaction with this para position, right? So reason is very clear. If you do the basic chemistry, you can easily give the reason why this is formed. And you can see here why it is formed. So after getting this here, right, getting the iodinium intermediate, and this is para is more reactive, that all of you know. So this attacks here, and then it releases aromatization, and this lead to. So whenever do any reaction, at the last, intermediate always try to get a more stable form, whether it is aromatic nature or whether it is a conjugated double bond or conjugated triple bond, right? And reaction goes in that direction. And once you have, look here, iodo handle. So this is known as Suzuki reaction. You can do the Suzuki reaction and you can have a variation at this end. And another reaction is called Heck reaction, is Nobel reaction, Nobel Prize winner, Heck reaction. So one can introduce this here, this alkene moieties here. And another is called Sonagasha coupling reaction. So this is the advantage of iodine catalyzed reaction. Both iodine is biodegradable, right? just water washing, whatever your reaction you do after that, just do the water washing and you can remove all inorganic things. And look here, right? So what is the next? Look here. Now this is another smart designing of the molecules. These are very, very used molecule. Indolo 1,2-equinoline. And you can find in several of the natural product. And what is a smart designing here? You know the pyrrole and indoles, they are electron rich at 2 position, right? Second position. So what is now this? If you have an indole and you can take iodo-bromo and you can do this CN coupling reaction, you can get this molecule. And after that, once you have iodo handle, you can introduce this alkyne by Sonagashia coupling reaction, right? Now, if you look here, this is in close proximity with this, right? So once you do the iodine will form iodo intermediate, and this 2 position of indole is electron rich, it can easily attack and you can get this molecule. So simply is use of the basic knowledge of the chemistry, nothing else, right? And how this reaction takes place as you can see here. Iodine will form iodine intermediate, then it will attack, and then aromatization that will lead to the molecule. And again, you have an iodo handle, right? And this iodo handle further you can functionalize, and this is confirmed by a single crystal, right? And you can see here, simple and useful chemistry, and you can see here the publications. Journal of Organic Chemistry, it's one of the core and leading journal in the field of organic chemistry. And when you read here in this journal, there's a lot of information is there on this. I have shown you just one reaction, right? But when you go to this paper, single paper, the supporting information is more than 150 pages. Supporting information means you will get all the spectras, right? That is more than 150 pages. So, and generalized chemistry is called when you can have a wide number of molecules, wide variation and large number of molecules, right? And once you have an iodo handle, you can do Suzuki, Hack, and annulation, means alkyne annulation you can do here in this, and you can make such kind of molecule. And when you have a polyaromatic compound, they are useful in materials chemistry, right? So now further you can do, once you have an iodo handle, you can hook up any nitrogen containing compound, fine. So, and now look here, this simple chemistry, right? We made 2 publications, and Tripti Agrawal, you know, she did this work, and she got the NOST is called National Organic Symposium Trust. This is in India. Every year they are giving 12 prizes, right? It was 2014. It was $1,500, right? And she got the NOST Best Thesis Award from Delhi University. She became a first from Delhi University who received this NOST Best Thesis Prize, right? So this is very prestigious thesis prize, and she did this work, simple and useful chemistry, and that is why she received this award. And now look here further how smartly you can design using a simple chemistry into a useful molecules. Diversity oriented cascade synthesis, 1 bullet more than 100 target. So by using this one, you can construct more than 100 scaffold. Scaffold means thousands of molecules, right? Now you can see here. So if you have, now you can see here, if you have orthoalkylaldehyde and another, you can take amine embedded with another nucleophile in that, right? So this is what is, I have shown you a retro by which you can have a 2, 3, 4, 5, 6, 7, 8 ring, and you can also have a chiral system. The only thing is that, why I am showing you only the one part of our lab chemistry. Simple and useful, because I can see here a lot of master students are there or PhD students. So what was the designing? Look here, how you can design. Designing, you can see here, this is orthoalkylaldehyde which we have used in the beginning. And you know aldehyde, when you react with amine, what you will get? Imine, right? So first is imine will form, right? After getting imine, now in this you have one nucleophile here, and nitrogen also has a lone pair, it can attack here. So question is, which attack will take place first, right? So which attack will take place first? This also need to be confirmed. So there is one thing, either this nucleophile can attack on imine, because imine, all of you know, is reactive. And imine is also reversible. When you have water, imine is also reversible, because the water can also attack. So there is a competition between water and the another nucleophile which is present, right? So this, either this can attack here and you can get a ring A first, and then the nitrogen lone pair can attack here, and then another route is this. Either this nitrogen lone pair can attack on alkyne, you can get ring B first, and after that this nucleophile can attack on this imine, and both will give you the same molecule, right? But how to confirm this? So this is the strategy by which we have made more than 100 scaffold, right? And these are the papers you can see here. Because there is no space, so I did not include it, the papers from 21, 22, 23. What I mean to say simply here, you need to use a basic chemistry and need to smartly design the molecules. Look here. So these are some of the papers, some of the chemistry which we have developed by using this, right? All these chemistry. And you can see here, these benzimidazoles are very, very useful, and some of these are also used as DNA intercalators, right? And by using this chemistry, you can see here first imine and then second intermolecular attack, and these are scaffolds you can easily synthesize by variation of ring A here, this side. Both the side you can vary, right? And further, the same chemistry now you can look here, we have extended for this. If you have this pyrrole phenylamine, right? First you will get imine, then again this will attack here. So one bond formation, second bond formation, third bond formation. There is no waste. All the reactant are used. Nothing is eliminated except the water, right? And all these scaffolds we have constructed using this chemistry. And so now you can see here, when we perform the reaction by controlling the reaction time. So when the reaction takes place for 2 hours, after half an hour, we have checked the TLC, and TLC we could find a different spot. So that we have isolated. Now you can see here, this clearly indicates the formation of A takes place first, ring A. So imine is attacked by a nucleophile, right? And the two different sets of the reaction, this is with benzimidazole, this is with aminopyrrole. In both the cases, we got this intermediate that clearly indicates ring A formation takes place first, which is clearly indicated in the single crystal, right? Now, another reaction you can see, this is in water. Water and ethanol, these are polar solvent. And polar solvent always play a significant role in driving the reaction. Because this is due to hydrogen bonding, they polarize the bond and makes that, generates the electrophilic center, right? So now look here. Further, we extended the same chemistry by using this, with having amine embedded with alcohol or amine, right? And this is with water and AgNO3, right? And again, similar chemistry, amine, and using this another component, and you can have a wide range of molecules, right? So you can see here, one can have these kind of starting substrate. You can vary according to your need, right? And then this is the another component, another part. And now you can see these molecules all are useful in biological point of view. It's not just synthesized. They have some significance. Now another reaction you can see here is, now you can see here all I am using either tandem or cascade, right? So continuous bond formation. And this is another set of this designing by which you can see here is 3-component reaction. So what you have is amine and this ketone, right? So, and this we have further extended for the sulfur analog of beta carboline. And now in this case, what you have, the product in which carbonyl group is present. So once you have a carbonyl group can be further explored by various reactions, right? So you can see here, we have further extended the substrate scope by using a different carbonyl group and amines. And this variety of molecules can be constructed, right? What I mean to say here, just I am showing, means how smartly you can utilize the basic chemistry. And that should become a generalized. Once generalized means it can generate a large number of molecules, then it is useful, right? So this is the mechanism here. First is enamine. And all of you know the enamine are also reactive, right? So once it will form an enamine, and simultaneously this aldehyde will react with amine and it will generate imine. Then enamine and amine will react again. And this is all takes places inside in situ, right? And then you can see here this can attack here, and after getting this enamine as a nucleophile will attack here. And then generation of this intermediate F. And the reaction can also take place directly. Either this can attack here and this can attack here. This can be confirmed through isolating the intermediate. Hydrolysis of enamine, all of you know, generate again the ketone and releases the amine part, right? And in this the proline was used. And further, can we use this chemistry for using the chiral compound, stereoselective compound? So now you can see here, we have extended this chemistry for making, starting with amino acids. Amino acids are not soluble in organic solvent. So initially, we tried this reaction by using amino acid. So it was not successful because of the solubility. Then we use amino esters, right? So amino esters, you can see here, we have used amino esters, and by using this amino esters and using gold, and this is where we have used ethanol, right? So this all wide variety of compound we have constructed by using this chemistry. And these kind of compounds, when you have a 1,5 oxygen, CO and OH, they are useful in as anti-HIV agent, anti-tumor. So we thought, can we use this chemistry like starting synthone for making such kind of compound? So we constructed this starting substrate, and with silver triplet in water, we have successfully constructed these kind of molecules. And the mechanism is very simple. Once you have this methylene, reactive methylene, you can easily remove the proton, right? And just simply by giving the heating also, you can remove that proton. And after removing that proton, right, it can form this enolic system and activated by silver, and this can easily attack. And this is how, this way or either this way, you can construct this molecule. So this is what is the substrate scope, further confirmed by a single crystal, right? So now you understand how smartly you can utilize this simple chemistry. And now you can see here, this chemistry we have used in 2009, right? And then we have extended this chemistry. You have aldehyde, and cyano group is equivalent to aldehyde, right? So we thought, can we replace this aldehyde with a cyano group, and can we construct directly amine containing molecules? Because amine containing molecules are very, very important for a biological point of view in terms of binding, right? So look here, we see the simple chemistry. If you have a nitromethane, what we have used? Simply KOH DMSO, nothing else. By using this will generate an anion and will attack on cyano, and this, you can see negative is there. And once you have a DMSO, so we also confirm from where this H comes. So when we use this deuterated DMSO D6, you got a D here, and still you have this acidic hydrogen. So base will remove this proton, anion is generated, and you can see here, this again will attack on this. It will take this D from here, and finally aromatization, this will lead to the molecule of NH2, or you can say ND2. So H is coming from DMSO. DMSO, dimethyl sulfoxide, is like acetone. Acetone has a reactive proton, and similarly here we have shown, right? So you can also introduce D here, right? So this is simply is an extension of the same chemistry and replacing the CHO with CN. And you can see here, large number of molecules we have constructed, and by making a different aromatic nucleus, functional group on alkynes. And also, if you have a base, one side you can construct this one, another side you can leave here, and another side also you can have a different variation in the group. And when you use 4 equivalent, right, 6 equivalent and 2 equivalent, both the side you can have a ring. In between you can have a aryl ring, right, and both the sides. So this kind of molecule could be used for material point of view. And now you can see here, these we have shown in this paper, right? These are representative because base and mono both can be used in this. Further, we have used another nucleophile here, you can see here, right? CN and acetonitrile, right? So once you use acetonitrile, CN is introduced. CN can be further used for various transformation, and you can use here instead of benzene ring, you can have a pyridine ring, and you can use acetophenone to introduce this keto group, right? So now this is a very generalized, and one can also use here, instead of alkyne, you can use alkene. You will have a dihydro compound, and you can use such kind of starting substrate to make these molecules, 5-membered ring. And also you can extend this for later stage modification in this natural product. So this is chemistry. And further, now you can look here. These are the molecules, natural product. So we thought, can we extend this chemistry for making directly for this kind of compounds? So what we have designed, look here. This is what we were using. Here what we have changed only, this benzene, CN and methyl group at ortho position. Once you have methyl group ortho to the cyano, it becomes a reactive. Cyano is electron withdrawing, that will make this acidic. So the proton is removed from here, and this will attack, and this here, right? And still you have acidic hydrogen here that will attack, and you will get amine. And amine will further attack on this cyano, so you can directly construct this. And further, if you are taking a DMF in excess, formylation can take place, right? So by using this chemistry, we have directly constructed these molecules. So this is further substrate scope. Look here, variety of molecules and natural product diversification. And further substrate scope of the same chemistry of this. And with alkene also you can further extend the chemistry. When you use excess of DMF, you can do direct formylation. It is a direct formation of the compound and introduction of this group, right? So there is no side product. And this is the mechanism of this reaction. Because of this electron withdrawing nature, this becomes acidic, that attacks on this. This is I have shown just now. And removal of this benzylic proton, it attacks here, and then you will get this intermediate C. C will aromatize to amine D. Amine D will again attack on this cyano, and that lead to the formation of amine containing this compound directly in one shot, and this is a natural product, right? So this is how you can construct. And further, if you use excess of DMF, further this next reaction is a radical reaction. You can do the formylation of NH2 group, right? So in summary, look here. In the last part, this can be used. So this we have replaced CHO with a cyano, right? And now look here, how beautifully this synthone can be used for making a variety of molecule where you have a NH2 group in the molecule, right? And when you have NH2 group in the molecule, when any drug goes inside your cells, you know, the binding is very, very important. So when you look inside your body, all is the chemistry. You know, the binding pockets. You have amino acids, right? And different kind of groups are present. So drug and medicines are designed in such a way so that they can bind at particular position. So this is what I have shown here. We did not use any metals except one position, right? So smartly how you can design your chemistry, and that lead to formation of a useful molecule. And you can see how many publications we have made out of this chemistry. Simple chemistry, sustainable chemistry, because cost effective and minimum waste, minimum number of steps, because in the single go you can have at least 5 bond formation, right? So in summary, this we have just shown you the regioselective tandem reaction, oxidative esterification, and cascade. So now you can look here outcome. I did not show one part here where we got patent on deuteration method when we started this one. And simplest and useful method for deuteration of organic molecule. We have filed only one patent and one is granted, right? So now you can look here, reward of the best publication. So these are our pillars from our lab. Tripti, she also got NOS Best Thesis Award, and she represented Lindo Nobel Laureate meet. And she became the first from Delhi University who participated in Lindo Nobel Laureate meet. And Lindo is a place in Germany. And after 3 years, the chemistry term comes. Every year, there is a different, the meeting with the Nobel laureate. There are always 25 Nobel laureate, 20 to 25 Nobel laureate, and 20 to 25 world leading scientists come from their field. And it is very competitive. From India, they select 25 candidates, 12 masters, and 12 to 15 research scholars, right? And in this meeting, they interact with the Nobel laureate one-to-one. And DST take care of all the expenses for this. And one need to apply for this. It's very competitive. And she got selected in 2014. And Rakesh is also the contributor from several chemistry. And currently, he got position in IIT BHU. Now, recently, he moved to IIT Roorkee. And he also he worked with Jonathan Clayton. All of you know, you are reading the books, right? And he also got a Marie Curie Fellowship 2019. And Monica, after this 2014, 2017, she got selected from Delhi University, from my lab, and she represented the Lindo Nobel Laureate meet. And now you can see these NOS Best Thesis Award, prestigious award, 17, 18, 19, 20, 21, continuously. And 22 was the COVID period, right? And in between, we did not nominated anyone. And Pawan is currently working in UK, and he also got Marie Curie in 2022 with 99.8 score. And this is, we always enjoy in our lab. So this is a recent picture of our alumni meet, right? So you can see here some of, and all are in same dress, right? Man in blue, woman in blue, right? And so these are our students currently, and you can see here the Rahul, right? He's from here, and he's so passionate. Whenever he's free from here, he'll end up in the lab. So this is what is needed. If you have to do research, the fire is needed. So he always comes whenever holiday is there, whenever he's free from after lunch, he goes there from here to North Campus. It's not easy. But only passionate people can do this thing. Right? And you know, when Shushmita was working in my lab, she was the first always reached in my lab from Dwarka to North Campus using scooty. And this is a picture is first alumni meet 2019. So these are all the pillars and who made this university and country proud. I proudly say these are the best students from our lab who made me possible to stand today here in between. right? And this is the building. You can see here inside. So this is here, I take the master's practicals here. So these are two opposite sides, lab 4 and lab 5, and this in the corner is chemistry. And this is a new building. It was designed for our chemistry, but unfortunately we could not get, some other people grabbed this thing. So thank you all of you. I have just shown all of you research comes from the passion and with a smart designing. You have seen, so because I'm from the university system, doesn't have in the beginning all the facilities. We were not having even the NMR in the starting. But we never gave up. So you need to have an attitude of like that. Never give up. Things will come automatically. Right? Work hard and use the basic knowledge. And I'm happy to see here you have a very good infrastructure. I'm sure very soon you will get all the other required instrument. Right? Thank you. Thank you all of you. If you have any question, please raise your hand. Any of you? Any of you, any questions? This is the problem. You never, ask the question even though when you are in the teaching class, very few people ask the question. Whenever you have a small query, you just keep quiet. आप लोग ये सोचते हो, just अरे मैं छोटा सा पूछूंगा तो दूसरे लोग क्या सोचेंगे, छोटा सा क्वेश्चन पूछ रहा है. Whatever you have any query in class, please ask the question with your teacher. Otherwise, that problem always remains for a long time. Because we are getting, if you look the plants, this comes from the plant photosynthesis, because plants are all green. We are getting the oxygen from there. If you look all over the world, everywhere, the majority of things you can see here, what we have is a plants. So that comes from there. Right? And plants, we have a natural product. We get the oxygen. We get so many medicines. Not only that oxygen. Majority of the natural product comes from there. If you look this medicine, right? Paclitaxel, anti-cancer compound, right? That comes from yew plant. And who is the inventor of that? This is Mansukh Wani, an Indian who's working in US. So inventor of this, paclitaxel is Mansukh Wani, an Indian. Then इसमें कुछ मैंने शो किया था that was different from there. But most of them I have shown you simply in iodine, right? So some reaction we always need. That's why people are always looking the alternate, right? And when you have a K positive, right? So K is there. When you use the water, it will dissolve in that, and you can remove, right? There are several metals, heavy metals, which are not dissolved. They remain in. And that's why we have to avoid those things. Yeah, you came in that. Good.
Those are really informative presentations. Thank you for it. I just have one question. You also talked about the synthesis of amino acids, right? So I couldn't help but wondering, how exactly are you stopping them from being viral? Because we know they can also undergo dimerization. So how exactly are you quenching them at that pace, or are you not quenching them?
In case of pyrrole?
In case of amino
we use amino esters. We didn't use amino acid because amino acid doesn't dissolve in water, so we use alternate method, we use amino esters. And an ester group later on can be hydrolyzed and you will get acid. And acid can be further used.
You talk about designing. So is it first theoretical designing and then testing in lab, or it is first testing in lab?
No. First, write on paper. Yes. First on your basic chemistry. You have to use your basic chemistry or basic knowledge. And look what molecule you are targeting, right? According to that molecule, you need to design. And design in such a way so that you should not use metal. And you have to design in such a way so that one group and another group are in close proximity, right? So that their next reaction, they can have a bond. So this is what is needed. See, once you sit and once you think, you will have several ideas, right? 10 ideas. Try to execute. At least one will work. Then design another one. If doesn't work, design another one. Definitely you will get success. And one need to design in such a way so that chemistry is generalized. In one chemistry, you can have several kind of molecules. Now you can see here, what we have designed, we got number of papers, not only one. Yeah, this is what is needed. But that takes your complete thinking, right? It comes from the heart. And you always need to think how you can make this, how you can think if it is not working, what should be the other alternate. Definitely solution will come. I know we have so many other administrative work and so many other things, but I am always keep thinking about the chemistry. And I am always in my lab most of the time. I enjoy teaching and chemistry in the lab. And whenever you have to grow in your life, whether it is chemistry, whether it is physics, or whether it is any other stream, you have to give everything from your heart. And then you will get success, 100%.
Thank you, Professor Verma, for your excellent insight into synthetic organic chemistry. Your expertise and clarity greatly enhance the understanding of the subject. It is an important information for the pharma and other industry. And I would request the coordinators to further. I request Professor Sanjeev Thakur to present the certificate and memento to Professor Verma.
So one more addition in
Before we take the session forward, we have our one of the guests here who has recently joined us, Dr. Gaurav Krishnan. He is a senior principal scientist at CSR laboratory. I request Professor Sanjeev Thakur to present a bouquet to sir. Ye raha, Uba. Bouquet. Welcome Dr. Gaurav Krishnan to the meeting. Now we all will assemble for the group photo because some of the members are leaving. हाँ, एक अच्छा अच्छा पहले हम लोग अरे पीछे
देख तो लो कभी कभी कमेंट भी करके देखती
अच्छा आगे हो ना Sir, lunch. नहीं मैम आप 1 मिनट में मैं डाउनलोड करती हूँ. नहीं मैं ये हटा रही हूँ, हट जाएगा.
एलईडी भी चला लें इसमें चला लें
इसी पे चलता रहेगा कोई.
कि वो बीच में जो सिग्नल छोड़ रहा है ना वो कितने सेकंड बाद छोड़ रहा है थोड़ा ठीक है.
उनसे पूछ लेती हूँ चलो. सर ठीक है मैम. आ रहा है.
ऑटोमेटिकली चेंज होते जाते हैं.
नहीं मैम कर रही हूँ.
तुम करते हो हाँ बैठ.
हाँ जी यही सर. अच्छा आई हूँ. आ क्यों नहीं रहे सर.
Our next speaker is Dr. Bhavesh Dave. He is the medicinal chemist with more than 18 years of experience in drug discovery and development. He has led projects from conceptualization stage to clinical trials in therapeutic areas like oncology, metabolic disorders, and immunology. Worked with leading pharma companies. He has an expert domain knowledge of medicinal chemistry and drug discovery. He has an experience of working with novel and established targets in different therapeutic areas and has represented medicinal chemistry department in a multidisciplinary drug discovery team to ensure success of the projects in international conferences. He has also supervised a number of PhD and MTech students. He is also a member of the editorial board of ISRN Pharmaceuticals International Pharmacological Journal Hindwani, and also serving as a reviewer in a number of journals. Dr. Dave, please.
Thank you everyone. Am I audible at the back?
first of all, I would like to thank organizers, HOD ma'am, and everyone at this university for having me here. It's really a privilege for me to be in front of students. It's more than 30 years since I was a student, and if I see this auditorium, I believe you guys are very lucky. Because at our time, we were not having such fancy setup. So cherish this and make sure you guys utilize this to the full extent. Okay, so before I start my talk, some general things that I would like to mention. I'm not a teacher, okay? I'm just a researcher working in the pharma industry. As ma'am mentioned, various pharma industries I've worked. And I'm not going to talk about catalysis here. I'm sorry for that. This is going to be a general talk on drug discovery. I'm sure you guys know a lot more about catalysis. So I thought I'll speak on something different, which could excite you to take up your careers in pharma industry going forward after your graduations, post-graduations, or your PhDs. So this is very general talk. Don't focus on slides. There is no rocket science here, okay? Just focus on the content that we are going to discuss, and let's make it a very interactive session, okay? If you hit a Google with drug discovery, you will get thousands of such slides. So don't worry about this. Just focus on what we are going to discuss, and ask me questions, okay? Anytime during the talk or after during the talk, I would like you guys to get engaged and start discussion, okay? This is all about discussion. So the topic is drug discovery. This is just an overview, just a bird's eye view because it's a long process. I'll come to that. So first thing that comes to you, anyone's mind is, why do we need drug discovery? Why do we need? Okay, let it come. Why do we need new drugs? There are already thousands of drugs in the market. Why do we need new drugs? Okay. So during COVID, we have all seen, earlier it was some other version, then Delta came, and then Omicron came, and then many other came, and it will keep coming. Not COVID, but many other diseases would keep coming. So diseases are, and the pathogens that cause the disease, they are very smart, okay? They need to survive. So for survival, they keep on changing, and due to changing nature of those pathogens, we need new drugs. Okay, so that's one part. Other part is unmet medical needs. Today we know maybe 100 or 200 diseases. Who knows, in future there would be 500 diseases or 1,000 diseases. So just to address those diseases, we need more drugs. And as I mentioned, diseases keep evolving. Resistance, you know, if we keep on taking antibiotics, sooner or later those antibiotics would not work in our body because our body gets resistant to those antibiotics. Bacteria, they are very smart. They overcome those drugs, okay? So for this thing, we need new drugs. Mutations. I'm not sure many of you would know more about mutations. So mutation is like we have proteins in our body, right? So in proteins is made up of many amino acids. So once amino acid changes, say from alanine to tyrosine, it's called as mutation. So what it does is it changes the complete structure of the protein. And drugs are no longer binding to that particular protein. They don't work. So we need new drugs. So this is mutation. New diseases, I already mentioned. Lifestyle changes. Okay, this is the age of technology. Sitting at one place, we get everything. So our lifestyles have changed. Our food habits have changed. So that leads to a lot of diseases. I don't know, you guys might have read in the news and something that
Nowadays, the cardiovascular events, the heart attacks and everything. Okay, so earlier that used to be in the older age, 55, 60 or above. But now the average age is 25. Right? So guys in the age of 25, 27, we had one colleague of 26 years of age, he was playing cricket, just collapsed on the pitch and died. So these lifestyle changes are making a lot of difference to our body, and this is leading to a lot of diseases. So we need new drugs. Okay, so in a nutshell, I've tried to explain that why do we need new drugs. Okay. So this is the overall process of drug discovery. Okay, there are many cartoons available on the internet, but last evening I made something. I thought I should not copy anything. Let's make something new. So I've tried to make this very simple cartoon, just follow the arrows. And each of the boxes that I have mentioned here, I'm going to talk about it in detail. So this is just a snapshot of how a drug discovery process looks like. Okay, first thing first is disease area, then target identification and validation. So by target, I mean a biological target where drug can go and bind and show the effects. Then you have to go for hit identification. You need to identify a chemical compound which can interact with the biological target and elicit the desired effects. Then hit to lead and lead optimization, you need to make the better compounds, the compound which is non-toxic, which is acceptable to the body. Okay? Then preclinical development, a very complex process, very resource intensive process, very expensive process. Then IND filing. IND filing is basically you prepare a dossier, and that dossier is submitted to regulatory agencies. In India, it's DCGI or CDSCO. Can we do something about this? Okay, so that's IND filing. Then once you get the approval, you can start your clinical trials. I'll come to clinical trials as well. There are various clinical trials, phase 1, phase 2, phase 3, where you test your compounds in human, okay? Earlier, all the preclinical development, your hit to lead and hit, that's all in animals or in vitro system. We can do that. So we were at clinical trials, right? I'll come to the details of clinical trials, what kind of clinical trials are there. Then comes the step of NDA filing. Like after successful completion of all the clinical trials, you again need to go to the regulatory agencies and file the NDA. So NDA is new drug application. Once you file that NDA, after approval of NDA, you can launch your drug in the market. So this is the basic workflow, you can say, of a drug discovery process. Now, there are two things. First is drug discovery, which gives you novel drugs, and second is generic drugs. You all might have heard about generic drugs. So generic drugs are basically drugs which were invented by someone else. They were patented. Their patent got expired, and anybody can make those drugs and sell it in the market. So those are already there. India is leading in that, as India is called the pharmacy of the world. So India is leading in that, but drug discovery, yes, India is coming up. Few success, but those are not significant. We need to do a lot more on those parts. But it's not that India is not doing anything. We have many companies which are working with MNCs of Western world and working shoulder by shoulder with them and giving out new drugs. Okay? Any questions on this slide? Please feel free. As I said, let's make it interactive. Okay. I'm not a teacher, so please feel free about it. Okay. Yes, please go ahead.
So you actually brought up a concept of launching new drugs. So I just had a question. As we know that we are specifically targeting all the mutated or the adapted pathogens to actually cure the diseases. Why can't we just modify the pre-existing drugs according to the mutation which we have? What are the challenges we can face in that?
You are absolutely right. We can definitely do that. So that process is called as drug repurposing. Okay, and it's been done world over, and we as a company, Sphera Pharma, we also have a technology platform where we are doing drug repurposing, and it's definitely can be done. You're right. But it has its own challenges. Sometimes things don't work as you expect. That's how science is all about, right? I have slides on that. So once that slides come, you guys can ask me. Yes, that's true. That's just a one sentence, but doing preclinical development, if it cost you 100 rupees, doing clinical trials would cost you 1 lakh rupees. Yes, there are a lot more. So when I come to my slides on clinical trials, you will understand, or preclinical development. There are a lot of regulatory things that changes with the time, okay? Things are a little bit easier till you do hit to lead optimization. Once you go beyond that, what is of the prime importance? The prime importance is safety, okay? Safety of the animal that you are dealing with, and later on it's safety of the humans that you are doing it. One single adverse event and your whole 10 years of work is going down the drain. No, no, it's not like that. It's a process. I'll come to that. So computational modeling is just a part of it. It's not everything. Okay, we consider computational modeling just as a tool. Okay, it's a guiding tool for us. But a lot of medicinal chemistry comes with experience. It's all about gut feeling. Okay. And of course, there is a rationale, there is a science behind it, but experience is the key here. Everything comes with experience. Okay, I'll operate. Just check if it's working. Okay, so we are at the first part, right? As I mentioned, disease area and target identification and validation. So what a company does is first it needs to decide like which disease area the company wants to work in. And this is purely a business decision, okay? Someone say that you can get X number of dollars in oncology. Someone say that you can get Y number of dollar in immunology and something like that. So this is purely a business decision where business leaders, they sit together and they take decision, okay, we are going to work in this area. So to do that, you need to do disease landscaping. That if I take example of oncology, oncology is not simple. There are thousands and thousands types of cancers, okay? So you need to see which one you are going to work on. What is the market for that drug? Suppose down the line you launch a drug for that, you make a drug, how much money it will get you? Like industry is all about money. They don't care about anything else, unfortunately. So once you decide, okay, that I'm going to work in this disease area, oncology is one, there's metabolic disorder like your obesity, diabetes, hypertension, then antimicrobials, your antibacterial, antifungal, antivirals. There are many, you name, there are rare diseases as well. So once you decide on that, then you go and look for the biological target. That suppose I'm going to treat lung cancer, for example. So in lung cancer, how can I treat lung cancer? So you need to find a biological target which is normally done by biologists. Okay, those who have experience in that, they browse through the literature, they find, okay, these are the 50 biological proteins are there, which I can target. You do a careful evaluation and you arrive at a target, okay, this is the protein I'm going to work on, right? So once that biological target scouting is done, you identify one protein, then the job starts, okay? First thing is you validate that target, that after manipulating that particular protein with any tool compound or genetic validation, or through gene knockouts or anything, whether it is giving the effect that I'm looking for or not. Okay, so that part is called as target validation, which is done using various techniques, like you can do proteomics, you can do genetics, or you can make some tool compounds, or as the gentleman mentioned, you can use some older drugs and see if it's working on that target or not, right? Then as a chemist, you would be interested, okay, what is my role in this process? What I'm going to do? So a chemist is normally a part of a target scouting team. So once they sit with biologist says that, okay, I have this 50 targets in mind. What do you think about it? So chemist has to see whether these are druggable or not, right? Whether you can make any drugs out of this or not, that's the job of a chemist. Chemist has to do that. And there are various modalities to target a protein. It's a small molecule which can go on and bind, but with that small molecule, there are 100 other things, okay? So you can do a lot of those things. So the second part is hit identification and then hit to lead optimization, okay? So what are the ways to identify a hit? So basically hit is a chemical compound which can go and interact with the protein and show some effect. We are not looking for a very great effect here, but yes, hit is a compound which gives us a little bit of activity in vitro. So what in vitro means? Something that is done outside the body of animal or human. Something that is done in a petri dish under the microscope or using various instruments, that is called as in vitro screening, okay? So the ways to identify a hit is the ones that I've listed here. HTS library, HTS is high throughput screening. So you take lakhs and lakhs of compounds. It's a robotic instrument that is there. So using that robotics, you can screen all those 1 lakh compounds or 10 lakh compounds and see which is the group that is showing the effect. You need to do clustering. Once the screening is done, results are out, then chemist has to sit through all those compounds and do the clustering. Okay, this is the particular chemical look-alike. Then comes the secondary screening for those compounds that you need to see whether they are false positives or not. Like, ऐसे ही तो कुछ effect नहीं दिखा रहा है. You need to make sure. So then comes the secondary screening with HTS. Fragment screening, again, fragments are basically, as the name suggests, they are very small molecules. Just molecular weight of 100, 150 or so, where using those fragments, later on you can build the molecules, right? So that is about fragment screening. Bias compound screening. This would be a little bit difficult for you guys to understand, but I'll try to explain key. Suppose you identify a target which is a kinase. So there are many types of biological targets. Kinase is one of them which does the phosphorylation and dephosphorylation. Okay, so there are libraries available in market in public domain which are kinase biased. So you can use the kinase biased compound library. Then DNA encoded library, this is a new thing which has started just a couple of years back. So you take the DNA out of the protein that you are looking for, conjugate some organic compounds to it and do the screening. AI ML based predictions, right? The in thing. Everything is AI and ML nowadays. So using AI and ML, our computer guys, they can make the algorithms, they can virtually suggest the compounds, and those compounds can be screened later on if one can make those compounds, right? Molecular modeling is another technique where softwares are available. So using those softwares, you can do the simulations and design compounds accordingly. Now comes the part, and as you can see, the star I have mentioned, the star is medicinal chemistry here. Thank you. See, this is what we do and this is what we love, medicinal chemistry, okay. So next couple of slides are medchem heavy. So this is where the major role is played by the medchem guys. So once you have the hit, which is shown a little bit of activity, now your next step is to optimize that to show more effect, right? That is always step one. So what you do in this is focused SAR. So SAR is structure-activity relationship. So once you have, say, 20 or 30 compounds made, you have initial data of those compounds. You need to make a sense of that data, okay? By using alcohol group, it is showing this much activity. If we have acid, it is showing this much activity. Amine is showing this much activity. Quinoline is like this. Naphthyridine is something like this. Thiazole is something else. So you need to make a sense of it. Okay, so that activity is called as structure-activity relationship. Next point, very important, fast synthesis of designed compounds. Okay, so this is the period where you need to make rapid progress. So to make rapid progress, your synthesis has to be really fast. Library synthesis. So library synthesis is like you make a scaffold and just go on and modify those scaffold within, say, short span of time, within few days or couple of weeks, you can make hundreds of compounds. These are all small scale compounds. You need only 10 to 20 milligrams initially. So at small scale synthesis, you can do, it's automated as well. There are synthesizers available in the market. One can use those synthesizers. And same goes with the purification of those compounds. Those also can be automated. And again, next point, very important. You need to establish IP. IP is intellectual property or what we call as patents. So in pharma industries, publication, nobody cares, okay? What matters is patent, because patent is the one that gives you the money. Publication doesn't give us the money. So we are always about patents. We don't care about publications. And patents also, see, I can make 10 compounds and I can publish a patent, no big deal. But if that patent gives me some product out of that, then only that patent matters. Okay. Like publications, patents are not peer reviewed. Nobody is going to review the patent you publish. But people can challenge your patent. Okay, the work that you are showing, someone has done that before. So before filing a patent, you need to do the prior art searches. You need to do the prior art analysis. You need to do the FTO, freedom to operate, and all those things that is taken care by the IPMG group, intellectual property group. Along with everything, you need to optimize your compounds for in vitro. In vitro, I already explained. That is something that is done outside the body. optimization of selectivity. So what selectivity means is, suppose your compound is hitting the target that you intend to. Along with that, it's also hitting many other proteins. Okay, if it hits many other proteins, it can lead to a lot of side effects, which we don't want. So you need to optimize your compound for selectively targeting your desired protein only. Then basic DMPK. So what DMPK is, drug metabolism and pharmacokinetics. So once we take drug inside our body doesn't like drugs, okay? It wants to throw it out. So it has many enzymes in the body whose jobs are to oxidize those compounds and clear them off the body. Okay, that's what we like, CYPs, cytochrome P450s. Those are the compounds. So basic DMPK you need to establish कि compound body में रहना चाहिए. If compound doesn't stay in the body, how it will show effect, right? So it has to show effect, it has to stay in the body. So little bit improvement you need to see. Then early in vivo efficacy in animal models. So first thing, it starts with animals. So you need to dose the compound in animals with disease intact or disease induced and see how the disease progression happens. Okay. So there are various techniques that can be used here. So suppose you are working on diabetes, okay? So you take a mouse or a rat, make it diabetic, ठीक है? You induce diabetes in that mice, you dose your compounds, and look how much sugar is lowered after dosing your compound. So these are some early readouts that you can do. Any questions here before I move on to next? For patentability, your compound has to be absolutely novel. Like, there should be no literature report of that compound. Patent protection is normally 20 years. After the publication of patents, your patent is there for 20 years, unless until someone challenges it. Okay. And there are ways and means to extend patent life beyond 20 years as well. You can file a lot of things. That's a separate lecture in itself. But yes, basically, if I answer your question, it's 20 years. Anybody else? No? Okay. Now comes lead optimization. Again, medicinal chemistry is the star here. Okay. First point is same. You need to do a lot of SAR, you need to make a lot of compounds here, okay? Unless until you make, you cannot screen those compounds. So synthesis is very important. And this is step further. Earlier we were dealing with say 10 mg, 20 mg, 50 milligram compound. But here you need gram scale synthesis, okay? So your synthetic route could change here. So when you make earlier compounds, you are not worried about the yield. Okay, I need the compound 10 mg. Doesn't matter if it's 20% yield, 40% yield, or 100% yield. But here it matters because you need compound in gram scale. So maybe you need a new route. So chemist has to work on newer synthetic route for that. Okay. Obviously purity, since this is going to be dosed in animals, so purity has to be very high with minimal amount of impurities in it. There are standard guidelines that one needs to follow. Purity in terms of HPLC, you can do HPLC, you can do NMR, you can do LCMS, and all those things are needed. You correctly need to assign the structure to the molecule that you have made. Okay, so those who are organic chemist and intend to make your career in chemistry, analytical chemistry is as important as organic chemistry. You need to be very proficient in NMRs, mass, IR, and everything. consider this as part of organic chemistry only. Okay. Optimization of in vitro potency, again, same thing, but you need to go places higher. So earlier, if you were having 50 or 100 nanomolar of IC50, so IC50 is inhibitory concentration. The concentration that is required to inhibit 50% of the effect. So if you were at 100 nanomolar, now you need to go below 1 nanomolar. So a lot of optimizations are needed here. Selectivity, again, if you were selective against 50, 60 targets, now you need to be selective against various types. You need to be absolute selective, okay. DMPK, preferably once a day dosing. So, एक बार ही dose करना है और the effect should last for whole day. So, medicinal chemist has to work on all this designing. So that's why I call medicinal chemistry as fun, okay. You get challenges every day, you get challenges throughout your career. So I give you one example. We were working on a collaborative project with MNC called Merck. You guys might have heard Merck, okay. So we were handed a novel target by Merck that you guys go and make drugs against it. So we made 500, 600 molecules. We got beautiful in vitro potency, less than 1 nanomolar. Beautiful SAR, but unfortunately, DMPK, where we were lacking. We could not get a good compound which was staying in the body, and we had to struggle a lot. But eventually we could do that, and it gave us a lot of satisfaction that yes, could do this. On slide, this looks very simple. ठीक है, चार point लिख दिया हो गया. But believe me, it's a years of job. As I mentioned, total drug discovery is 10 years, so a lot of time goes in this. You need to make a lot of compounds. So organic chemistry is the backbone. You guys should be very, very proficient in organic chemistry. Organic chemistry is our bread and butter. Without organic chemistry, can't do any of this, okay? That's the most important aspect. Then in vivo efficacy, you need to do in various animal models. You need to make sure that before you are hitting humans, you should be sure that, okay, my compound is going to work. To get that much surety, you need to test your compound in various animal models. And this is cost intensive as well as resource intensive. It takes lot out, okay. Combination studies, you need to dose more than one compound. You need to check effect of your compound with established drugs. Okay, because more often we see that a lot of us are taking more than one compound. So what is the synergistic effect of this thing? You need to see all that. Early toxicity readout, as I mentioned, from here on toxicity matters. A single episode of toxicity and years of your work goes in drain. It's completely non-negotiable. This is about preclinical development. So before you hit humans, these are some of the steps that you need to do. So now you have a compound which has shown good in vitro effect, which has shown good in vivo effect. It is selective. It works in animal models. What next? So what next is all this, okay? Process chemistry. Establish scalable synthetic route. So from milligram, we went on to gram scale, say less than 10 grams. Now you need to go beyond that, okay, say 50 gram, 100 gram, 200 gram, 500 gram scale synthesis. And it has to be as per ICH guidelines. So what is ICH is International Council of Harmonization. So few scientists, they got together with regulatory agencies all over the world, and they rolled out a process and criteria that any drug you are taking to humans, it has to meet certain basic criteria. So those are called as ICH. Identification of metabolites and synthesis if needed. So what are metabolites? So as I mentioned, that body doesn't like the drugs. It wants to throw it out. So in order to throw it out, it basically destroys the molecule. So what fragments you get, those fragments are called as metabolites. Okay. So if needed, we have to synthesize this as well. Then synthesis of radioactive labeled compound. Once you finalize a compound, you need to make radio label where one part is deuterium and second synthesis is carbon-13 labeled synthesis. Okay, so deuterium anybody can do in their labs, but carbon-13 is done at specific places only. Everybody doesn't get licensed to work on because it requires a special setup. So BARC in Bombay, BARC in Bombay, that has approval to make C13 labeled compounds. Deuterium you guys can also make in your labs, not a difficult task. Extensive DMPK studies, okay, you need to do the mass balance. Suppose आप 5 mg compound body में दे रहे हो, so what happens to those 5 mgs? You need to take account of it, okay? So lot of extensive studies you need to do ki what is the effect of food, whether you should take your drug before food or after food. ठीक है, डॉक्टर कहते हैं ना हमें कि खाने के पहले ले लो या खाने के बाद ले लो. So all that is driven by the DMPK studies that we do. Formulation development. So what is formulation? The compound that we make is called as API, active pharmaceutical ingredient. ठीक है? So that API we just cannot pop up like that. It has to be formulated in a pill or a capsule or a syrup. Okay, so that is called as formulation. So we need to do all that. Non-GLP talks. So what is GLP? GLP is good laboratory practices. So the labs which are GLP certified, they have a certain setup, they need to do certain documentation. So first thing you need to do is non-GLP talks because GLP talks is very expensive. So to cut your cost, first do some initial non-GLP talks in your facility only, and then you can go to GLP certified labs where you can outsource your work and they can do the work for you. CMC established. CMC is chemistry manufacturing controls. So now we are talking about manufacturing. Okay, so it has to be on kg scale, ton scale, and beyond that. So that plants or pilot plants needs to be established, everything. It's a big process that needs to be done. You need to do commercial scale synthetic route. So there are special people who have experience in production, who have experience in process chemistry. So they do all the commercial scale synthetic route. Okay. Plant scale manufacturing in FDA approved. So FDA is US FDA, Food and Drugs Administration. So they visit various plants and based on their setup, they give the approval. So once a drug is made in US FDA approved plant, it can be sold in US. If a drug is not made in FDA approved plant, it cannot be sold in US. And US is the major market, right? Strong QA, QC, quality assurance, quality control. Okay, so those are guys, we call them as inspectors, police inspectors. So they check each and everything, whether you are following the right process or you are not following the right process. If you are not following the right process, they suggest you the remedial measures. That has to be followed. Then comes the GLP talks in 2 species. 2 species is one rodent and non-rodent. Rodent could be mice or rat. Non-rodent is generally a dog or a monkey in rare cases. So those are basically 2 cycles you have to do, 30-day dosing and a 90-day dosing. That's the minimum. Regulatory agency may ask you to do even more, and they normally ask you to do more. Stability studies, the compound that you have made, whether it's stable or not, the formulation that you have made, whether it is stable or not. You need to do stability studies, say at 25 degrees, at 40 degrees, under different humidity conditions that you may normally encounter while storing your medicines. Any questions here? I'll take a pause. I'm not used to speaking so much, guys. Sure. So they're very different. Only thing common in between is dosing in the animal. Okay. So in DMPK, as the name suggests, it's drug metabolism and pharmacokinetics. That is basically what body does to your drug. Okay. Once we take the medicine, what our body does to that medicine is DMPK. And what medicine does to our body, that's in vivo efficacy. Okay, as I mentioned, I gave the example of diabetes. Okay, after taking a diabetes drug, how much my blood sugar is lowering? That is called as in vivo efficacy. So that is the basic difference between the two. Two species means one rodent and one non-rodent. So rodent could be mice or rat, and non-rodent is normally dog or monkey in rare cases. Yes. Two rodent and one non-rodent. So dog is normally must. People do monkey also sometimes, and rat and mice. Anybody else? Now comes the next step, that is called as IND filing. So what is IND? IND is investigational new drug. It's new drug, but still investigational because it hasn't cleared the human clinical trials. So you need to do drafting of the document. Okay, that is a specialized job, special people do that. QA, QC reports, inspectors come into picture again. Okay. Documentation, I'm sure you guys are writing your notebooks once you do practical in your lab, right? So same thing. Here also, anybody who is working needs to document specifically. Procedure has to be written specifically, well and clear. It has to be signed and cross-signed, right? Your analysis, NMR, IR, mass, HPLC, everything has to be in place. Pre-IND meeting with regulatory agencies. So regulatory agencies are the agencies which will approve your drug or your IND. So you need to go and meet them before you file the IND. Tell them that this is what I'm intending to do. What are your views on that? So they tell you that, okay, you need to do this, 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 you need to do this. So under their guidance, you need to draft your package and work accordingly. Clinical trial planning, discussion with clinical CROs. Anybody cannot do clinical trials. These are special setups. I'll come towards that, what is those, but those are called as clinical CROs, custom research organizations. You need to work with them. Drug substance supply, again, as I mentioned, whether it's a capsule or a pill or a syrup, or it's IV dosing, or it's parenteral like injection or anything, whatever is there, company has to ensure that enough supply is there of the material. Clinical trials. So clinical trials, basically, there are three types, phase 1, phase 2, and phase 3. So phase 1 is done in healthy volunteers. So people like us who are healthy, okay? except for oncology. For oncology, world over, there is permission to do phase 1 trial directly in the cancer patients, because cancer patients are really sick. They need access to the medicine very quickly. So for that, world over, agencies has given a leeway that if your drug is for cancer, you can directly dose in the patients. So what they do in phase 1? So phase 1 is normally 25 or 30 volunteers at a particular dose. Once you increase your dose, then numbers increases. So after dosing the drug, they take out the blood and measure how much drug is there in the body at, say, after 2 hours, after 4 hours, after 8 hours, 16 hours. You don't need a hospital setting for this. Clinical CRO can do this thing, right? And by doing this, you establish what is the safe dose for my phase 2 trial. Phase 2 trials are going to be in patients. Okay, so by doing this, you establish how much drug can I give. ठीक है, 5 mg की कैप्सूल देनी है कि 10 mg की कैप्सूल देनी है, कौन सा देना है, right? Then comes phase 2 trials. So this is where you see whether your drug is working in humans or not. Okay, so suppose a person has a cancer, you give your drug to the patient, you monitor them for 6-7 months or 12 months and see how much improvement is he getting in cancer. या तो cancer कम हो रहा है, whether it's going down or it is staying same or it's increasing. Those are normally done. Same thing with all the other diseases, right? And this is normally limited number of patients, say 200 to 500 patients. You can do it at couple of sites, right? Blinded trials with placebo. So placebo is nothing but control, dummy, yes, very rightly mentioned. So it doesn't do anything. So the doctor who is giving the drug to the patient doesn't know whether this has the drug or this does not have the drug. So that's why he is blinded. The doctor is blinded here, right? So it gives more control to the clinical trial. There is no biasness here. Phase 3, large scale you need to do in multiple countries, multiple sites, across multiple ethnicities, multiple places, different genders, ethnicity, everything matters here. Because if you are planning to sell your drug world over, there are different populations. My body is not same as your body, right? We are different. So it needs to be tested in various places. So this is a humongous task, okay? This is very intensive, phase 3 trials. And once drug comes out of phase 3, then you can say, yes, I have a drug. After that, during this process as well, you continuously keep updating your data to the regulatory agencies who are going to approve your drug, that this is what I'm getting after dosing the patients. There are independent committees, okay, they also go through your data. They check your data very thoroughly, whether it's working or not working. There is no biasism here. It's strictly scientific evidence. And all these trials are done under very strict rules and regulations. Okay, every government world over, they have their own set of rules which needs to be followed. Patient consent is mandatory, their video monitoring is mandatory, adverse event reporting is mandatory. Even if there is a single adverse event, it raises a red flag, okay? So ultimately, when expert committee decides on approval of the drug, they go by risk-benefit ratio. Okay, there is no drug which has no side effect. Every drug has one or the other side effect. But whether that effect is better or side effect is more detrimental, that's what they decide. So now comes the final point, okay? Once you are done with everything, your clinical trials are done, now is the time to launch the drug. So as I said, expert committee reviews your data. After approval of data, company plans for the commercial launch. Okay, that's when the role of project managers come into picture, product managers. They devise the marketing, how you are going to market your drug. You need to reach out to doctors, explain them everything, how this is working, tell them benefits of your drug, and that's how the marketing strategy is planned. Any questions so far? Then I'll come to the career slide. Yes. Yes, we did. We did. But so what was done during Corona is it was given accelerated approval as investigational only. Because you had the patients already at that time with you. You need not search for patients. And it was a global health emergency, right? So during that period, vaccine was developed quickly and it was administered. Yes. No, they did follow, but for a shorter period of time. Suppose normally vaccine development is say 4 or 5 years of process. Okay, not like small molecule. Small molecule takes a lot of time, but vaccine doesn't take that much time. So suppose your phase 1 trial is for say 45 days or 60 days. Instead of that, it was done in say 15 days or 25 days, something like that. I don't know exactly what procedures were followed, but as far as I know, all the procedures were followed, but they were shortened just to make it accessible to people because it was a global health emergency. But everything was followed in that case. And as far as side effects are concerned, a drug like paracetamol, okay? Paracetamol we take, that also has a side effect. So there is no drug which doesn't have a side effect. It's only the risk and benefit ratio. So those vaccines saved thousands and lakhs of lives, so beneficial. That's my personal views, okay? Yep. Enzymatic is in vitro part that is done outside the body. And in vivo is done inside the body. DMPK is also inside the body. As I explained here, okay, so DMPK is what body does to your drug and in vivo efficacy is what drug does to your body. That's the basic difference between two. In vitro is everything outside body. In vitro is nothing inside the body. It's done in petri dish and microscope and all that. That's a science in itself, okay? So based on the properties of your molecule, the disease you are going to target, uske base pe decide hota hai. It's a time-consuming process. And there is a specialized branch called as pharmaceutics. Okay, the guys who do pharmaceutics, they are experts in formulation. It's done in B Farm and M Farm. Yes. See, I'm not an expert in homeopathy. I can't answer your question on that. I'm sorry for that. Yes, you need to do all that. It's not mandatory, but just as I mentioned during start of my talk, like all computational approaches are basically guidelines. So after doing docking, you come up with say 50 molecules. And then you start making those 50 molecules based on your gut feeling that, okay, I like this molecule better than the other one, so I'll make this molecule first and the other one next. No, for that you don't need to do any docking. Okay, docking is done when you are targeting a protein. In case of radiotherapy, you are basically treating whole body. So you cannot do whole body docking, right? And there is for the radiotherapy or phototherapy, there is no particular protein or particular receptor that you are targeting, right? Yes. Not only the cancer cell, the whole organ you are targeting. No, there are organs and tissues in everybody. Whether person is lean or obese, it is always there. So in, say, as I mentioned, that my body is different than your body. Okay, the absorption of drug could be different. Suppose you are taking paracetamol, I am taking paracetamol. For you, it can work in 15 minutes. For me, it can work in 30 minutes. Okay, so that difference will always be there. So in case of phase 1 clinical trials, okay, which is done in healthy patients, okay, so once a drug is dosed, basically what clinicians do, they see what are the levels of the drug in body. Okay, to treat a cancer, I need certain amount of concentrations all throughout the day. Whether I am getting that concentrations all throughout the day, that's what is mentioned there. Yes. Yes, humidity does affect stability of drugs in certain cases, and while doing formulations, those things are taken care of. That's why normally in any strip or any bottle of drug, storage conditions are mentioned, that you need to store them at 25 degrees in a cupboard, or in case of vaccines, you need to store them under refrigerated conditions, right? So for HIV, drugs are there now, okay, but people struggled a lot to produce the drugs for HIV because the virus that's causing the HIV, it's very heterogeneous in nature. Okay, it's very difficult to target that virus with a single drug. So HIV nowadays treatment is a combination of 2 or 3 drugs. So because of the complexity in virus, it was challenging. Okay, I come to the slide here where I define career opportunities for chemistry students in pharma. So this is just about pharma industry. I'm not going to talk about any teaching or research institutions. Okay, because I'm not an expert in that domain, so I'll talk what I know. So first thing first, as Akhilesh also mentioned in his talk, that you need to be very, very strong in your subject. We interview many candidates, okay, day in, day out, but I'm sorry to say the quality is not there now. We really struggle to select a good candidate who has basic knowledge of organic chemistry. Organic chemistry means you should know the structures, you should know the name reactions, you should know the mechanisms, you should know the NMR, mass, all those things you should be knowing. Okay, you need to be very strong in reagents. So if you want to make your career in pharma industry in organic synthesis, make sure your organic chemistry is very, very strong. Okay. So once you apply to a company, learn about that company, what they do and what is your aspiration, what you want to do in that company, okay? You can apply in various pharmaceutical companies, drug discovery companies, or CROs or CDMOs. So pharmaceutical company is basically an umbrella. They do each and everything that I mentioned here. Okay, so be sure what you want to get into it. You want to get into organic synthesis, or you want to go into analytical as instrumentation. You want to be QA, QC persons. You want to go into formulations, intellectual property. You need to learn the WIPO courses, okay? Business development is also a field where a science graduate can take up that. But you need a little bit of experience as well. So you need to spend a couple of years on the bench, and after that, you can take up the role of business development also. Project management, after I think you can do project management courses after our post-graduation as well. Okay, those are called as PMP certifications. So if you do PMP certification, but for that also you need some basic experience. So you need, after your master's or your PhD is over, and if you want to work in industry, go into the industry and get some experience, okay? And I tell you what we face challenge with the freshers, okay? So when a fresher joins us, first year goes into training, okay? And after getting some train, they leave. They go and join some other company for just 5,000-10,000 more bucks. So what we have practically done is we have wasted 1 year of ours. So that's the challenge we face with freshers. So make sure you enter into the company, stay there for a few years. I don't say that you stay there for the lifetime, but stay there for meaningful time, because 1 year is very short time to learn organic chemistry, right? But make sure that you utilize your time, you work very hard. So in industry, chemistry labs never close at 6 p.m. Okay. You need to work very hard. There are basic minimum criteria as part of your KRS that are set. So typically every day people need to do 3 reactions and 2 purifications. That's very challenging. But that's the bare minimum criteria that needs to be done. Or if you are a tech-savvy guy, go and learn data science, go and learn AI, ML, computational modeling. There are fields open for that as well. You can do that. Learn about intellectual property, as I said, patents, and try with law firms. There are many law firms in Delhi and Gurgaon. Okay, you can try. They also recruit freshers, by the way. So if you are interested in patents, then that is the side you can go. So this is what came to my mind. You guys have any questions, please feel free. Internship programs for chemistry is a little difficult because lab safety, as Akhilesh mentioned during initial talk, lab safety and confidentiality. So because of that, in chemistry lab, internship is difficult, but you can take internship in other domains like intellectual property or some data science. The company, those are dealing in data science, you can take internship opportunities there. Some companies do offer internship for chemistry, but many don't do. We don't do at Sphera. Inorganic and physical students, they can definitely get into formulations. Okay, but it's tough. It's better to do pharmaceutics. Analytical also they can go. There are many inorganic chemistry specialists who are part of analytical. And if you have strong knowledge in organic synthesis, then that also is fine. While recruiting, we don't see whether you are from organic or physical or inorganic. What we need to see is the basic knowledge. How much knowledge do you have about chemistry? That's what I said. Data science, AI, ML, you can try. Yes. So, there are many law firms who prosecute patents of the pharma companies. So they need guys who have knowledge of chemistry. Okay, because pharma industry is majorly about chemistry. So if you want to get into that, do some WIPO courses, WIPO. You can do the internet search. There are some free courses available. You can do those courses and then approach them. Yeah. How am I doing on time-wise? I'm done? Okay. Thank you, guys.
Thank you, Dr. Bhavish Dave, for your enlightening lecture on the drug industry and guiding the students about the pharma industry. It is an area where students can make a bright future. So I would now request Dr. Sonawale to present the certificate and memento to Dr. Bhavish Dave. Ma'am, chalo.
Please feel to reach out to me anytime. Even if you guys want to visit our company, most welcome. Just let us know 2 days in advance so that we can make some.
Industry
Such a knowledgeable session it was really, and we thank Professor Anjana Sarkar for chairing the session. Now I request Dr. Nazmul to come forward and present token of regard to Professor Anjana Sarkar. We have lunch ready outside, so we'll assemble here in 30 minutes back after lunch. Yeah. Everyone, please. Our guests, our eminent guests are requested to join for lunch here. This side we have lunch. For the students kindly join for lunch this side. And for our guest dignitaries, we have lunch this side. Students are requested to join lunch this side. On your right. Yes, please be back here in 30 minutes. Students, kindly assemble here in 30 minutes. I request our guests to please move forward for lunch. This side, na? Uma? next presentation. take your seats faster, settle faster. Silence, please. Yes, so this time after lunch is a bit probably lethargic for you, but that shouldn't be the case here because we're going to have very important and very amazing presentations here which will give you updates about the recent focus of the research. So we have our guest who has recently joined us, who has joined us before some time, Professor Ram Singh. Let us welcome him, and I request Dr. Sunita, if she's here, I request Dr. Leena to come forward and present a bouquet to Professor Ram Singh. Now I invite Dr. Sushmita to session next chair, next session. Sushmita? Yeah. Thank you.
Good afternoon all. In the session 2, we are having 2 invited lectures, lecture 3 and 4. For the lecture 3, we are having Professor Ram Singh from Delhi Technical University. The title of his presentation is Harnessing Catalysis for Green Organic Synthesis. Professor Ram Singh received his PhD from Department of Chemistry, University of Delhi in 2003. After that, he carried out his postdoc research at the Center of Environmental Management of Degradation Ecosystem, University of Delhi. And presently, he is a professor at Department of Applied Chemistry at Delhi Technical University. The area of his research focuses on organic synthesis, natural process, product synthesis, and chemical biology. And currently, he is having more than 103 publications in the leading organic chemistry journals. I welcome you, sir.
Thank you.
So, good afternoon. It's challenging to have a lecture just after lunch. So, first of all, I'd like to So I'm thankful to the organizers for inviting me here. And the topic that is, I think, chosen or given to me, it's in between both of that. That is harnessing catalysis for green organic synthesis. So keeping in mind that this is our after lunch first lecture, I will try to keep it simple. And I will be covering 2 parts of that, as the title suggests. चलता नहीं इसमें? I think LED पे चलेगा नहीं ये? नहीं चलेगा ना? ये चलेगा. ठीक है. So, catalysis and green organic synthesis. So, first of all, why we are so much concerned about that, the word letter green. And then I will go for the catalysis. And last few slides, whatever a little bit I am contributing towards that. So all organic synthesis somehow, somewhere they are using catalyst. And their parameter is something different than what physical chemists look for the catalyst or inorganic chemists look for a catalyst. So I will be covering basically, as I told you, that the green part, and green and sustainability goes side by side. Some of the historical aspect I will give, that why things have changed, what was the reason. So we will be talking about the sustainability, then why green or green chemistry, what impact catalysis has on that, how green chemistry is different from environmental chemistry, or is it same? Then something about why catalysis is green, then we can go for some of the examples. Sustainability, we all know that whatever we are doing today, are we keeping the same thing for tomorrow? Tomorrow means our next generations. Obviously, we don't want that the generations to come will have a cylinder on the back, mask on the face, and they move around. Though, due to one or other reasons, we faced with the mask. If you will see the condition of human activity, these are the two pictures of Delhi. Lockdown has a lot of ifs and buts, but if you will see from the point of view of the environmental aspect, there was so many examples that has been published in that time that due to the lockdown, things has become better, environment has become better. So obviously we want the next part rather than the first part. If very frequently in Delhi you have the grab 1, 2, 3, about the pollution that parameters is being imposed. The vehicles having the BS3 petrol, BS4 diesels, they are not being allowed. Just to make the things easier for us. So our life depends upon the three things. We have air, water, land, and the question comes, are we doing justice to that? What human activity is involved in all that? So sustainability, obviously we are not following the protocols. In the previous lectures, we have shifted from industry to academic, obviously. That previous lecture was totally based on industry. And they also talk a lot about regulations. In academics also, we follow the regulations. For the environmental impact, we have a lot of guidelines. Even those teachers who are involved in the NAC accreditation and other things, they have a column. And we as an institute, we feel something in that we are doing this and that for the labs and other things. So this remains unsustainable environment we are producing. Then what is the solution basically? If you will see the subset, because we require economy, we require society, we require environment, all the three things is important for us. If something is covering all the three, that is the sustainability. And this is a challenge. If you go for economics, we are losing the environment. If we go for the environment, we are losing the society. So all three has to be taken care of. So we have to follow the three overlapping circle model given by the sustainability people, those who are dealing with the sustainability basically. So what is the solution, ma'am? Solution is we should stop the human activity. That is not possible. Now what is the possibility? We have to go with the alternatives. And here the role of young people like you starts. If you will talk about us, we have already spent 2/3 and 1/3 is left. And depending upon if you will see the world record of the global records, after 45 or 50, to survive is a difficult aspect, and various aspects are there where the numbers are decreasing. So you have to think the alternative, what alternative you can start from now onwards, so that when you will reach at the average of 35, 40, you will have something better than what we have today. Chemistry has an important role to play in achieving a sustainable civilization on Earth. This has been said. And why this has been said? Because nothing is without chemistry. Our recent subject, revision or other things, that is being given by the government or some other, I'm not commenting basically on UGC, ICT, but still, new education policy, we are removing chemistry somehow from the first year engineering branches. And I can say that this will have some adverse impact after 10, 15 years. Because no engineering branch can move ahead without the chemistry. If you will take even the computers, IT, software, they are dependent on the laptops, computers, and they are all materials. And without chemistry, materials cannot be there. So, and when you are going for synthesis of all these things, you require the methods where the role of catalysis comes. So is chemistry help us to achieve sustainable environment? What is the answer? Obviously, the total control is not possible, but yes, you can reduce. Even if you are reducing something, you are helping the environment for the future. So, there comes the term green chemistry. If you'll go into the history of that why this word has been coined, what was the requirement, you have to go back to 1960s. There was a book called Silent Spring. And this scientific book came in 1962, which outlined the devastation that certain chemicals had on local ecosystem. One of the examples that you must be reading in your school days, that is the DDT and pelican birds. Birds are one of the best indicator of the environment. So, that was one example. That book contains so many examples. If you get a hold of this book, do read it. So, in 1970, then the US President, he established US Environmental Protection Agency, EPA. That is a regulatory agency which is devoted solely to protecting human health and the environment. So the first major decision that was taken was the ban DDT and other chemical pesticides. There's a long list. If I go into the EPA website, you will find all the lists are given there. In 1980s, the chemical industry and the EPA were focused on pollution, cleanup toxins. They start searching something which will reduce or remove the pollutions. The industry people, the government people, the agencies, they sit together and they started getting some points. Based on that, they formulated some guidelines. So in 1980s, the establishment of green chemistry took place, and the two gentlemen, Anastas and Warner, they co-authored the book Green Chemistry, Theory and Practice. In 1998, that book came, and that book discussed about the 12 principles of green chemistry that you must be reading right from your first year onwards. In 2000, Green Chemistry Institute became a part of the American Chemical Society. And later on, one of the best, what you do, all ifs and means all the labors, all the works, what you require is a Nobel Prize for.
Sir, this is what at least I'm teaching to my students, that although in 1998 a book came into the picture and these two scientists brought these principles of green chemistry, but they were in practice previously also. It's just that they have coined all the 12 principles together, and then they have made a series out of it, but they existed previously also. Is that correct?
Yes. I will come to that, one more slide when I will talk about the catalysis. That whatever things are there, it is not that the things are not there. A simple example you can take carbon nanotube. Carbon nanotubes, the people isolate from that, ये बहुत साफ है. वो घर में जो वो लगता है ना जाला, मकड़ा का, उसको एक्सट्रैक्ट करना, उसमें बहुत अच्छे कार्बन नैनोट्यूब मिलते हैं. तो ऐसा नहीं है कि कार्बन नैनोट्यूब डिस्कवरी होने के बाद मकड़ी ने बनाना शुरू किया. Not that. It was there. Another example you can take, I will tell you later on. That structure I will show you. So, things are there. What you're requiring is the documenting the things in a proper manner and enforcing it. That is important. Surplus epoxidation, they got the Nobel Prize. Surplus has got Nobel Prize twice. Recently also he got. So these are the 12 principles of green chemistry. And if you see closely, all the principles are correlated. So out of these 12 principles, if you will see the role of catalyst, they are the 1, 6, and 9. That catalyst, prevention of waste or byproduct, I will come to it, all the three. Design for energy efficiency and use of a catalyst. So what the 12 principles they gave, these three are getting impacted with the catalysis, if you are working on the field of catalysis. What Dr. Bhavesh was talking about, the analytical tools, see the point number 11. So if you will see, I will just first 6 and 9. I think you have, so many examples are available for other principles you can find in the literature. So it is better to prevent waste than to treat or clean up waste after it is formed. And we have, prevention is better than cure, that is known to us since from our ancestors, that we should prevent it rather than treat it. What this says that, suppose I'm taking a simple example from a class 11th book, that there is a first equation that we read is CH4 with the chlorine, we are getting 4 products. Each hydrogen is getting removed. You have a CHCl3, CH2Cl2, CHCl3 and CCl4. Suppose tomorrow you are going for setting up an industry and you end up, you want to prepare a chloroform or CCl4. Now if you will follow that principle of that reaction, methane plus chlorine, you will get 4 products. You require only 1 product for commercialization. So 3 product is waste for you. Right? So you have to search the method or the route which will minimize the production of that 3 product and maximize the yield production of CCl4. That's why the percentage yield has been replaced by atom economy. If you see, I will go in the back, the point number 2. Now in most of the publications, you will find that they have given atom economy, not the percentage yield, or they give both. So green chemistry has a separate lecture itself. So here, see, whole of the chemistry is revolving around your reactant, product, and the reagent. You have to control only the 3 things. And chemistry doing, writing in the paper, chemistry doing in round bottom flask is entirely different. It's very easy to write on the paper. When you stand up in the lab, take a round bottom flask, you perform a reaction, A plus B will never give you C. They will give you A plus B is equal to C plus D plus E plus plus plus. Whether it is 0.1% or 9.9%, doesn't matter. If you link with the previous lecture, purity comes here, role plays. Suppose your medicine is having a 5% impurity. कोई भी डॉक्टर सुबह दोपहर शाम आपको खिलाता रहेगा, आप ठीक नहीं होगे. तो सुबह दोपहर शाम जब वो खिलाता है उसमें आपकी प्योरिटी 99.99% से ज़्यादा होना चाहिए, तभी वो ड्रग जाकर के आपको हेल्प करेगा. उसके बाद भी उसमें साइड इफेक्ट होता है. राइट? सो यू नीड अ प्योरिटी. For that, you need a purification process. Purification process is more difficult than doing a reaction. You can do reaction very easily. That's why it is always recommended that you read 4 days, if in a week you put up 1 reaction. So you read for 5 days, put a reaction, then take 2 weeks to purify it. So if you will give A plus B, give you C plus D plus E plus other things, so you have to design the reagent, whether it is a reagent, whether it's a catalyst, that will minimize the D, E or any other product. Where this is the place where role of catalyst comes. That is the thinking point. Just one or two examples I have taken from the literature. A multi-component one-pot synthesis has been coined up because of that one only to minimize the waste. Otherwise, the stepwise reaction always have each step you are purifying it. Second was number 6, design for energy efficiency. Suppose a reaction is being performed at 240 degree or 500 degree centigrade, 400 degree centigrade, and by your change of the reagent or catalyst or even the starting material, if you are bringing down the temperature to 100, 115, you are almost doing something good for the sustainability. So, saving energy. We all are talking a lot about energy, and next 15, 20 years, research is going to be on energy only. Let me tell you. We are shifting from traditional coal to alternate forms of energy. Electric vehicles is coming up, but it has a very big problem in future. Battery रखोगे कहाँ? जब वो useless हो गया, then what you will do with that? Exactly, right? So, today we have a lot of electronic gadget as a e-waste, as a big challenge to tackle with that. And now we are moving towards the developed country. So we are not the dumping ground. Earlier, what will say that developed country dump things to us. Now we are slowly and slowly opposing that. But we also have to dump somewhere. It's another big, very good field. If you can work on isolation of lithium from lithium-ion batteries, if you have a proposal, give it to Ministry of New and MNRE, New and Renewable Energy. You will get funded for that project. So, photo energy, you have photochemical reactions, microwave chemistry, sonication, and use of catalyst. Microwave chemistry is a very interesting chemistry, started very good, got lot of publication, but unfortunately, it does not reach to the commercial scale. As the previous speaker said that when you require things in milligrams, you can use anything. But when you require things in tons, you cannot follow the same protocol. Use of catalysis itself that we are discussing here, that catalytic reagents, they are superior to stoichiometric reagents. Then you prefer biocatalysis. Catalyst to be of high turnover number, means one catalyst can be used so many times. So what is the outcome of green chemistry? Just 3 points I discussed. Green chemistry not a solution to all the environmental problem, but yes, most of the fundamental approach to preventing pollution and give a sustainable life. It helps you in reducing the things at source. Just one slide for the difference of environment chemistry and green chemistry. Apart from other differences, I will focus on only one point, that there are 12 rules for the green chemistry and there is no rule for environment chemistry. But if you will ask me personally, in my opinion, environment chemistry is a set, then you can call it green chemistry as a subset. Discussions are going on that this is our, this is yours, that is different case. But this can be included in environment chemistry also. There is whatever point green chemistry additional has. So why is catalysis green? If it is following 3 principles of green chemistry, obviously this has to be a green. So this reduces your energy, amount of reagents, your byproduct, and when it controls byproduct, it will control the waste also. Waste management is again a taking up field where you can commercially viable field. When I am talking about commercially viable, that means you set up any startup which will benefit you. If you happen to go with this Chandigarh highway, Panipat refinery, if you sometimes have time, go and visit, or any refinery, sulfur is lying down, so many. NTPC, fly ash. Even that parali, every now and then, apart from the political statements, think about the science on the parali. Agro waste. Search catalyst that can convert that agro waste into value added products. कभी भी आपके घर के अगल बगल वो मार्केट लगती है वीकली, 10 बजे के बाद कभी एक चक्कर लगा लेना. You will find the amount of agro waste getting generated from there. Think how can you utilize that? Because landfill is already, whatever we have in Delhi, they are saturated. New lands yet to be acquired or whatever be the reason. If you will happen to go with this Valaswa dairy side, you will find that the mountain, though this is coming down now. Okhla, there is a waste to energy plant. If you have an industrial visit type, you can go and visit. So, exactly. But that can be included in environment also, environment chemistry. You can call it, yes, fine. That, you can say to that. But if I go in some other aspect, why we give a particular, so I'm taking one example. We have a lock and key method. We have a molecular recognition. We have a supramolecular chemistry, right? If you will look at closely, they are almost similar. Lock and key, particular lock, there is a key. Like molecular recognition, one molecule is recognizing another molecule, is like you have a ATGC, right? Same if you will go with the supramolecular chemistry, you have non-covalent interactions, which is there in earlier also. So sometimes when we coin a new name, the focus becomes higher. Right? Even in your clothes or in anything, if it comes with the new name, we at least for the initial 50-20 years, we give importance to that. So yeah, you're right, what said is right. So, but catalyst is not the answer of all the things. Still, it has a lot of disadvantages. So, separation of a catalyst residue from product, recycling of the catalyst, degradation of the catalyst, toxicity of a catalyst. So, these you have to take care of. Not that just because of the using a catalyst, if a reaction product or your atom economy or energy reduction is taking place, you cannot ignore the toxicity. All heavy metals, a particular amount is recommended for your body. The moment that exceeds, you will be in trouble. But still, in general, it is greener to use catalyst than not to use it. So again, I will link with your previous lecture. They say that there are side effects, but as long as it is helping more, you can opt for it. So if you will classify, we all know these classifications, homogeneous, heterogeneous, biocatalysis. These are some of the examples where a lot of catalyst is being used. You see the metals being used, apart from these zeolites. You have a zeolite, you have a montamolite clay, you have a silica, you have alumina. They are even better as compared to the metal catalysts. And nowadays, the non-noble metal catalysts are getting more preferences than the noble metal catalysts. You have alumina, silica, zeolites, they are available. They can be modified as acidic, as basic. So just to take one or two case study that how catalyst is useful for a synthesis. This was the initial synthesis of ibuprofen. Ibuprofen is a medicine you all must be knowing. हम खा लेते हैं दुकान में जाकर के. Earlier it was synthesized in 1, 2, 3, 4, 5, 6 steps. Each step you have to purify, then you have to go further. For purification you have to use lot of solvents, time consuming, loss in that percentage yield decreases. If you will compare the yield with respect to the first starting material, you will come at very low yield in the last product. When this steps has been utilized with the help of catalyst, it reduces to 3 steps. This was the Presidential Green Chemistry Challenge awarded to BHC company, which is now known as Base Corporation. They designed that 6 steps into the 3 steps. Another important point here is consumption of CO. Any process where you are utilizing carbon dioxide, carbon monoxide, it is itself a green. So that is the beauty of catalyst that we are using it. Next case study is use of zeolite. 225 degree, as I told you, energy saving. 225 degree coming down to 100 degree. So again, this is following the principles of green chemistry, and you will call it a catalyst used for this process is green in nature. Synthesis of paraxylene. Paraxylene is a starting material for PET bottles. We know how much you are using. So normal method, we have both ortho, meta, para, but with the use of zeolite, because of the site selectivity, the space, they allow only the paraxylene to pass through. And this ortho and meta is being rejected by this method. So it is better to use catalyst in this type of reactions, then you can go with the normal method. So mode of action of a catalyst, what required is the activity, selectivity, and stability. It must be active enough, selective. This is the example of selectivity that is given here, that they are selecting out of the 3 products. Stability means when they decompose after their use or during the course of reaction, they will be stable enough, and as per definition of a catalyst, they has to come out as such. So activity of a catalyst depends upon the texture, electronic structures. Activity of a catalyst, this you can explain with the help of active center, geometry of the surface, electronic structures, formation of surface intermediates. And for stability, you can say chemical stability, thermal and mechanical stability. So if they are qualifying all these parameters, you can call it as a good catalyst. Now how this important role plays by this catalyst, you can always judge with the help of the number of publications coming out. In last 10 years, almost 10, 11 years, this non-noble metal catalyst, as I said you, that their importance is more in comparison to the noble metal catalyst, because noble metals, they are the heavy metals, again, their toxicity has their own problems. So 8,000 number of publications, almost 8,000 means in 11 years is coming to be around about monthly, 85 publications monthly on one particular topic. So you can take up these small projects when you are opting for your project works. Biocatalysis, the similar type of numbers are there, that about 1200 and similar type like having 100 publications per month is coming on biocatalysis. Not only the publications, even the global market. If you go for the economics aspect of that, 31.1 billion market size in 2023, this has been recorded of this catalyst. And that we have a Asia Pacific market. India is there. So being in India, the chances is very bright that we can take up the role of catalyst for organic synthesis. So journey so far, yeah, as she was mentioning that earlier it is not there. We all know that the art of producing alcohol from sugar is known since the human history. That means biocatalysis was known at that time also, though we were not knowing the name. The first known use of inorganic catalyst is from 1552, when sulfuric acid to catalyze conversion of alcohol to ether. Some few examples I have taken. In 1781, the potato starch mixed with distilled water and cream of tartar, that is acquire a sweet taste. But when this was replaced with the acetic acid, the reaction become faster. So, though the word catalysis has been given in 1835, but before that also their role was there. And obviously the recognition in the field of catalysis, you will find that the Nobel Prize given in 1909, then 1918, then 1963, Ziegler-Natta, we all know the use and applicability of Ziegler-Natta catalyst, whose nowadays, if you will, 6th and 7th generation is being utilized in the industry, polymer industries. 1973, 2010, 2021, organocatalyst. So a lot of Nobel Prizes given in the field. That means the field has a lot of scope to work. And people are caring for the environment with the use of catalyst. So this is about the background. Some of the few examples, whatever I have done, though my main focus is towards the neurodegenerative diseases, looking for natural products, secondary metabolites, and herbal formulations. And waste management is imposed by my university to work, being in applied chemistry field. So I will take one example of my PhD time. I think Dr. Uma will appreciate this molecule. Very well-known molecule from Professor Chauhan's lab. So whenever we enter into the lab, we have to work on this molecule. So when I entered into the lab, I find that our seniors has worked with the help of HCl, acetic acid, and boric acid. So I thought that why not? Obviously, the purpose was the publication. You have in the previous lecture that patent was the focus of industry, but in academics still we have publication is the focus. Now things are changing. We are going towards the patent because again, this is a requirement of the regulatory body. When your institute go for the NAC, they ask that how many patents you have filed in the last 5 years. So we are shifting. So this isoloxagen, this is a core moiety for vitamin B2, riboflavin. And this cyclocondensation reaction, it was tried with the help of solid acid, montamalite, clay, alumina, silica, zeolites. Then use of this in the solution phase as well as microwave. And fortunately, I got 2 publications from that. So that served the purpose of myself and my supervisor both. These are just the series of the molecules. Obviously, if you will find the percentage yield compare, at that time there was nothing like atom economy, so much popular. This is about, I think, 2000. Otherwise, I would have calculated atom economy also. List of molecules, you can find that the percentage yield is drastically increasing and time is getting decreased. There were a lot of publications coming at that time from microwave. Every lab was using that microwave. The other important molecule of our lab was porphyrin. So this was, I was saying that I will talk later. This porphyrin is a very molecule known for since 1800. But there is another molecule called N-confused porphyrin. So after working for more than 100 years, I think the first publication or people came to know in 1994. Not that earlier it was not there. Now why we miss this type of product? Suppose you are in the lab, you perform a reaction, you know that when you are purifying from the column, that this is your product. So your first priority to get that product. What is there in below, what is there in above in the column, we don't care. We just throw out. But if you will patiently run the column and you can separate your D, E, F for academic point of view, I am dead sure that you will get a publication. We usually throw that. So not that the N-confused porphyrin was not forming earlier. It was forming earlier also with the synthesis of porphyrin. But nobody was caring that. We have a very purple color band in column chromatography. We separate it very fast, go for the next step, complete the chapter, get the PhD, I am happy, everyone is happy. But if you have a patience, isolate all the byproducts. You will get very new type of chemistry. Later on you can optimize. This is another paper of Journal of Heterocyclic Chemistry where the highly functionalized pyridine being synthesized. Why I synthesized all these molecules? Because my purpose is to link two molecules from the NH2 side. Again, that molecule I got through the screening of molecular docking, what was talking before the lunch lecture. So I am synthesizing those molecules and targeting the two diseases, Parkinson's as well as Alzheimer. So in due course of the synthesis, we use sometimes the catalyst. This is a recent publication. Even 2 days before I got the acceptance of this paper. This is again using the bimetallic catalyst nanoparticles. Then some of the natural product plants that are working is Rubia cordifolia, Arnavia nobilis, Phygopyrum, Esculetum, Bombec siva, your bell patra. I am using the bell patra from the temples that we are using on Shivratri. I thought I have seen so many bell patra after the Shivratri it is being thrown. So I asked my student to collect all those bell patra from nearby temples. Until now I got 2 publications out of that. These are the past scholars. These are presently working under me. And my research output is having the articles around 100 or so. Authored 16 books, book chapters, and e-modules on EPG Pathshala. You will find so many modules written by me. Symposia, conferences, and have granted 2 patents, one on Alzheimer, one on water dispenser. So thank you to my funding agency, DST, CSIR, DRDO, and some of the industries. They are also giving me little about of amount, 2 lakhs, 3 lakhs. I am happy with that. So thank you. You all are welcome for any type of support regarding the research field. If I know, I will support you. Thank you. Anything you want to ask? या तो सभी क्लियर था या कुछ भी क्लियर नहीं था. चलो, थैंक यू.
Wonderful presentation, sir. I would like to invite Professor Purnima Jain to present a memento to Professor Ram Singh as a token of regard. Our last speaker of this lecture series is Dr. Gaurav Krishnan. He is working in an innovation protection unit in CSIR as a senior principal scientist and handling patent application filing and maintenance. He is also a member of different committees in IPU pertaining to CIASC, etc. I welcome you, sir.
इससे होगा सर आगे पीछे सर सर ये सर तो नहीं जाएगा
Good afternoon to all of you. I think you have enjoyed good lunch, and post-lunch sessions are very much difficult to handle with without sleep. So all are ready. I also enjoyed a very good lecture by Professor Ram and earlier speaker, Dr. Dave. Some of the lectures are very good, and some students have just pointed out for this patenting and all. So I think this session might be very interactive, okay? And trust me, one thing I would like to ensure you ki I will make it very simple, okay? If you feel that anything is going tougher, then just stop me at that particular moment, okay? So before starting IPR or patenting, I would like to ask to you people ki how many students or faculty known about the patenting and how much patent you have filed. Please raise your hands. Nobody? Okay. So I'm very much, I should put it up to very lower. So as per the Government of India directives, in 2016, there's an IPR policy launched by the Government of India, central government, where vision and missions are written like that, ki Indian intellectual property led to growth in creativity and innovation is encouraged for the benefit of all in India intellectual property. And the mission like that to establish a vibrant and balanced IP system. Like these are the policies, documents, and I have just written down all the IPR policy, whatever the matters written in the vision and mission document. But here I would like to discuss some IP ecosystem in academic system. I am just like to add this one, IP ecosystem, where we start something like thinking from ideation to patent certificate, where you people have think something, your idea is generated, then it will convert to the patent application, then patent process is going on, and after filing you can get the patent certificate, okay? So have you think about this IP ecosystem inside your organization? Anything like that? कभी सोचा है ऐसा कुछ जहाँ आइडिया जनरेट हो फिर उसके बाद हम कुछ उसका करें ओके सो आई एम जस्ट एडिंग दिस की हाउ दिस IPR policy helps us to derive this type of IP ecosystem. The topic is a little bit dry, but I am trying to make it more interactive. So after this IPR policy, our patenting system become more faster. Like yesterday, I have just read the news that we have filed 90,000 patent in a year, okay? And 75,000 patent has been granted in last 10 months only. It's a record high patents. 75,000 patent has been granted in last 10 months only. In India only, in India, one particular. Actually, there are territorial rights. Patents are territorial rights. जहाँ हम फाइल करेंगे वहीं ग्रांट होगा और वहाँ पे उसका प्रोटेक्शन हमको वहीं मिलता है ठीक है कंट्री वाइज मैम, यस, टेरिटोरियल उस कंट्री में फाइल करेंगे, इंडिया का है, स्टेट वाइज नहीं होता कुछ भी. So we will see one by one. First of all, definition, very simple definition of intellectual property. What are the intellectual property? The something which are created with our minds. जो कुछ भी हम सोचते हैं ना, कुछ सोचा, उसका कुछ बनाया, that's our intangible assets. Tangible assets we have, like assets, whatever money, we are just saving it, we are just keeping safe lock and bank, all the things. But whatever the intangible assets, like IP, whatever the generated inside our minds, that's all called intellectual property rights, literary work, artistic work, design symbols, and the rights which are secured, that's all called intellectual property rights. Okay, very simple definition. What are the IP are? After that, type of IP are two broad category. One is copyright, that is protected in artistic work, literary, dramatic, and the second category consists of the patent, industrial property, that consists patent, design, trademark, GI, trade secret, integrated circuit and layout design, plant protection and varieties. These all comprises the industrial property, and first is the copyright. Two broad categories are there. Okay? As we discuss, patents or any IP is governed by the particular act, that are the act written like Copyright Act 1957, governed by the, that is copyright governed by this act under Ministry of Commerce and Industry, DIPP. Patent are governed by the Patent Act 1970, Department of Industrial Policy and Promotion, Ministry of Commerce and Industry. Design as governed by Design Act. Similarly, trademark are governed by the Trademark Act. And GI, geographical indication, that's for the particular geographical location that mark is given by them. That is also by DIPP. And the semiconductor integrated circuit layout design by DIPP, and the protection of plant variety under this one, under Ministry of Agriculture. This is a, you can say, a type of act, and what are the ministries related thereof. So we are discussing about the patent only here, because all the IPs are not possible here to discuss, and a lot of criteria are there, only patent. Like Indian Patent Act, since patent are the territorial rights in India, patents are, we can file the patents as per the Indian patent law, and that would be governed by the Patent Act. The first patent law was passed in 1856, and also known as the first Patent Act, Act 4 1856. Then first amendment was going on 1872, and the third amendment was in Patent Act in 1911, and presently we are handling the patent cases of Patent Act 1970, that was amended again in 2005. So we read it like the Patent Act 1970 amended on 2005, and Rule Patent Rule 2003. So right now we are following it, last one only. And all the rules and regulations for patenting activity, we are taking from 2005 amendment onwards. So some historical background is necessary, like what is the patent? The patent's word is originated from the Latin word, which means to lay open. And the first patent was systematically granted in Venice, in Italy, in 1450 for a period of 10 years. Earlier it was 10 years, but right now it's life varies from, life is 20 years fixed, okay, from date of filing only. And the first US patent was granted in July 31, 1790. These are some historical things we should know about before starting something. And the first Indian patent was granted in 1860. I think 1856 is the first one engineering patent. We will discuss this. And some information regarding why we need to patent product. So obviously, powerful tool for socio-economic and cultural growth of a country, to foster the innovation, to get the financial benefit, and balance with the innovators and the wider public interest. And whenever we got the monopoly for particular invention, then obviously somebody makes some better innovation for that one. So these are some points why we need for the patent only. in that continuation, it is very strong to make the market position and competitive advantage. Research, return on investment. ROI means return on investment. Then inventor can get the income from licensing and assigning it. Access the technology through cross-licensing and image building of a company, like earlier our speaker said, for any organization, patents are the patent portfolio has a huge impact of the industry. Means how many patents they have filed, these are the brand value for their company. And powerful tool to take the action against imitators. Like for anybody is infringing that, you can make a patent so you can stop them. So after this type, some points, we should share this some patent-related information in deep, like patents, which are administered by the Patent Act 1970, Patent Rule 2003, which are recently amended in 2005 and Rule 2003, okay? These are the exclusive right and territorial monopoly right for the particular country. Like we have to, if we had filed the patent in India, then we have the monopoly rights in India only. It is for 20 years, okay? Right now it is for 20 years from the date of filing. And granted by the government for the invention right for new inventive and industrial applicable process and product. We will discuss what are the criteria of patents. Then who can file the patent? Who are the applicant? Who are the inventor? As per the Section 6 of the Patent Act, any natural person can file the patent. He can be a assignee, a legal entity, a startup, a small entity. They are the assignee of the invention. And the true and first inventor can be anybody who are not the first importer of an invention. Alone or jointly, you can file the patent. And the legal representative of any deceased applicant can file the patent. So, like the applicant has disease expired, then his or her legally representative can file the patent. These are the some points for the jurisdiction only. If the applicant reside in this, these are the map of India, and the northern portion, northern part of the country, northern resident can file the patent in Delhi. Southern part can file the patent in Chennai, western can file in Mumbai, and rest of the India can file in Kolkata only. We have to file in particular jurisdiction only. The residence address that is decided on the jurisdiction decided on that address written by the inventor or applicant. So after that, some historical background regarding the engineering patent. The first engineering patent filed on February 28, 1856 by Government of India for the Pankha polling machine. On March 3, it has been granted. Pankha polling machine, you know, this is a drawing for Pankha polling machine. This is very not so much clear. To George and Dampening, he is the inventor. In February 28, 1856, a Pankha polling machine has for the patent for this one has been granted in India in Calcutta Patent Office, by Calcutta Patent Office. Earlier it was we have not the AC-like system. Na? A Pankha is there. We used to pull that one. Na? We have seen in TV earlier Raja Maharaja use करते थे कुछ ऐसे. उस machine का उन्होंने patent लिया था 1856 में. It was supposed to be the first engineering patent in India. So this is the basic question जो आप लोगों ने पूछा था कि scope of patentability and the patent, what are the patentability criteria, ठीक है? Criteria कौन-कौन से होंगे? So we will discuss here. The main point is like that. First of all, your invention your work should be in the form of invention. आपका work, your work should be in the form of invention. कोई भी चीज़ patentable तब होगा वो पहले invention होना चाहिए. What are the definition of invention? Under section 21J, Invention means the product that is a new product or process involving an inventive step and capable of industrial application. That should be invention. कोई आइडिया जनरेट हुआ तो उसका हम पेटेंट सीधे नहीं ले सकते, पहले वो प्रोसेस या प्रोडक्ट के फॉर्म में होना चाहिए, ठीक है, और आफ्टर दैट हैज़ इन्वॉल्व एन इन्वेंटिव स्टेप एंड कैपेबल ऑफ इंडस्ट्रियल एप्लीकेशन. कैसे होगा वो हम अभी देखेंगे. तो फर्स्ट वन इज द इन्वेंशन, वो इन्वेंशन होना चाहिए. Invention, then that must relate to a process or product or both, and must be novel. Novel क्या होगा? नया होगा. कैसे होगा नया? How it be new? It shouldn't be disclosed anywhere in any public domain. Public domain में वो publish नहीं होना चाहिए कहीं भी. Involves an inventive step. Inventive step, आप have you know anything about inventive step idea? We will discuss here. And must be capable of industrial application. That your invention must be capable of industrial application. Industry में भी use हो सकता हो, ठीक है? And not fall under section 3 and 4. Patent Act के section 3 and 4 में नहीं आना चाहिए. 1, 2, 3, 4, 5. 5 criteria that should be fulfilled. Yes, that is the important 5 mandatory criteria for the patentability. We will see one by one. That should be must be invention, relate to a process or product or both. That should be novel, that has an inventive step, capable of industrial application, and not fall under section 3 and 4. First one is the novelty. What is novelty? An invention is considered as new if it is not anticipated by prior art publication, prior use or prior public knowledge. An invention is new or novel if it has not been disclosed in the prior art. Before date of filing, it shouldn't be published anywhere. कहीं भी आपको prior art search में वो दिखना नहीं चाहिए. Then it should be novel. You have to ensure for the novelty first. There are some exceptions also, we will discuss. Then inventive step. Inventive step is the feature of an invention that involves a technical advancement as compared to the existing knowledge or having economic significance or both. It's a very simple, I am just telling ki, like we are using this bottle, na? So earlier we are using this bottle which have the problem of leakage. Like अगर उल्टा करते थे तो उसमें पानी गिरता था, लीकेज होता था. But right now, this is leak proof, है ना? This is due to this lock system. It's a very good example and simple example of inventive step. Means कि whatever the existing bottle was there, but we have introduced this lock system and become more useful. And this is leak proof right now. So it's a very good example of inventive step. And the second one, invention not be obvious to a person ordinary skill that art. Ordinary skill, कोई बंदा उसको बना ना सके. So I think nobody can make it in a simple, very simple definition. The second definition I would like to introduce here, like a very simple example for Hunt patent. Hunt was the inventor who has just invented a pin, a simple pin which are using to clip that cloths, okay? But earlier, what are the problem with this? In 1849, he has invented this pin. But the problem was this pin that it is used to, it is not very comfortable, okay? So, it is just pinching the body, it is just taught the cloths. So, pin becomes safety pin by introducing this one, this angular size, shape, and these type of shapes. This is a very good example of inventive step. Pin becomes safety pin. And it is more useful and you can say more economical and more significant, having more economic value. By introducing this type of angle type structure, the corner of this pin. So it's a very good example of inventive step. So then whenever you are thinking for an invention, you have to prove for your inventive step also. It is very subjective matter. I am just telling the key only for this pin example. How can you show like that? Yes, yes, yes. This, oh, sorry. This is already patented, qualified, this invention. And industrial application, already that is, you have, that invention must be able to give any industrial product also. Some of the, you can say, we had discussed the novelty, na? Some of the sections, like section 29 to 35, deals with the anticipation rules, which are not destroying the novelty of your invention. Like we are discussing some project here in public domain, means learned societies, exhibition. So according to section 29, if you had published anything which is, anybody has published without your consent, and if you submit, communicate to the government for any funding agency or anything, your project is carrying on, and you have just submit some project proposal to the government, then you are just disclosing your idea, na? And if you are displaying that your ideas before the learned society, and it is displaying for the public working, and you have to publish after provisional filing, then these are not considered as the anticipation. मस ये नॉवेल्टी डिस्ट्रॉय नहीं करेगा अगर आप 12 महीने के अंदर फाइल कर देते हैं, 12 मंथ्स के अंदर उसका एप्लीकेशन फाइल कर देते हैं. सो इफ just note down, if any type of this events happen, you have to file your application within the 1 year, 12 months. These are the very, suppose like you feel that ki something has been disclosed with your friends or in any exhibition, then you have to file that case, either provisional, within 12 months. So under section 29 to 35, anticipation, you have to keep these 5 points in your mind. You have to file the case within 12 months. ठीक है? हाँ, yes. Design solution, design, you can register the design separately, ma'am. Design is the different wing. Patents, design, trademark. For any type of design thing, you have to file the design only. Register the design. Okay? The design is a different IP. We have discussed like this. See, in seconds, both pen can, you can file the design of the pen and product also, patent also. So suppose if I am not interested in making a product, I can just. Yes, you can file the design also. Even in this particular mic, you can see, you can file the patent also. You can design 1,000 of you in your mobile phone. Na, you have 1,000 patent, 1,000 patent. Like this one, na, ma'am. Design, the second one, industrial property. Patent, design, trademark, GI, trade secret, integrated circuit, and plant protection. These are the. So we had completed the anticipation. Then which cannot be patentable, the fifth one, very important point. These are the patentability criteria, but which cannot be patentable. That comes under section 3 and 4 of Patent Act. What are the section 3 and 4? If anybody is feeling something problematic, then just stop me and ask. ठीक है यहाँ तक? वो simple तरीके से मैं बता रहा हूँ. Okay? Then what are the section 3 and 4? Section 3 and 4 deals with the not patentable subject matter, where under section 3 and 4 said that an invention which is frivolous or which claims anything obvious contrary to the well-established natural law, like a machine giving more than 100% performance, any perpetual kind of machine is not possible, na? So that is not patentable. Or anything that is Newton's law of gravitation, if you want to patent it, you can't go. Section 3A said. Section 3B, something against the morality, like if you have made something machine which is for making the bomb, you can't make a patent for that one. Under section 3B, that is, you can't make. You can't file the patent. Any gambling machine device for housebreaking, these are not patentable subject matter. Okay? This is written in the Patent Act, okay? 3A and 3B, then 3C, mere discovery of a scientific principle or formulation of an abstract theory, like Archimedes' principle or like that is already established. You can't get a patent for that, and don't try to file a patent, okay? Section 3D, mere discovery of new use of known thing, like I'm using this mic now, like I'm just saying ki I'm using this mic as a paperweight. I'm using like a paperweight. We can't get a patent for that. Known चीज का नया use हम नहीं ले सकते. Yeah, I'm using pen, but that is for writing only, and I'm just thinking कि pen is used for the paperweight or paper clip. We can't get a patent because it is a new use of a known thing. That is not patentable. 3E, a substance obtained by mere admixturing the result for aggregation of the properties of the component of the process for producing such a substance. This is not patentable also. Only we have to prove the synergistic effect for that one. And 3F are very good for engineering candidates, like a mere arrangement or rearrangement of duplication of known devices. Like we have an umbrella and torch, two devices which are working independently properly, but if you combine both the umbrella and torch, and we are just saying this is a new device and working properly, we can't get a patent because it is falls under section 3F, that is mere arrangement and rearrangement. If you want to get a patent, then we have to prove that the complete technical effect should be unique, okay? We have to show the technical effect for that particular device. So 3F is very much relevant to the engineering students. 3G, a method of agriculture or horticulture, a new form of plant, improved soil, that are not patentable, 3G. 3H, any process for the medicinal, surgical, curative process, diagnostic process, that are not patentable, 3G. कोई डॉक्टर किसी ऑपरेशन को करता है तो उसका पेटेंट नहीं ले सकता. डॉक्टरों के पास वो अगर डिवाइस बनाता है तो उसका पेटेंट ले सकता है. कोई मेडिकल डिवाइस है तो उसका पेटेंट ले सकता है. बट 3H इज रिफर ओनली फॉर द मेडिसिनल. कोई सर्जरी करता है या कोई ऐसा चीज़ जो कि इसमें आता है 3H में, तो वो नहीं कर सकता. 3I deals with plant and animals in whole or any parts thereof or any microorganism that includes seeds, varieties, species that are not patentable. 3I. And section 4 deals with the invention related to the atomic energy. सेक्शन 4 हमने जो भी एटॉमिक एनर्जी से रिलेटेड है, रेडिएशन से रिलेटेड है, उसमें पेटेंटेबल नहीं है. ये 4 आ गया था उधर. 3J, एनी मैथमेटिकल और बिजनेस मेथड और कंप्यूटर प्रोग्राम पर से और एल्गोरिथम इज नॉट पेटेंटेबल इन इंडिया. कोई कंप्यूटर प्रोग्राम बनाया और आप सोच रहे हैं कि उसका हम पेटेंट ले लें इंडिया में, तो नहीं हो सकता. दैट्स आर नॉट पेटेंटेबल. वी हैव टू मेक एन सिस्टम, हार्डवेयर एंड कंप्यूटर प्रोग्राम इज एम्बेडेड, मीन्स कोई हार्डवेयर का मिलेगा, सिस्टम का मिलेगा जिससे वो चलता हो प्रॉपर, उसका पेटेंट ले सकते हैं. बट एज फॉर कंप्यूटर प्रोग्राम दैट्स आर नॉट पेटेंटेबल. एल्गोरिथम पेटेंटेबल नहीं होगी, बट यूएस में है, ईपी में है. एज अ सॉफ्टवेयर वी आर मेकिंग एन एम्बेडेड सिस्टम. वेबसाइट नहीं कहेंगे, आप कहो कि कोई उसको एज सच वेबसाइट पे भी नहीं मिलेगा, सिस्टम पे मिलेगा. ऐप मतलब समथिंग विच इज़ रिलेटेड टू हार्डवेयर. कोई मशीन चली, कोई डिवाइस चल रही है, तब मिलेगा. वो लंबा चौड़ा क्लेम लिखना पड़ता है उसका, मैडम. ठीक है. एंड सेक्शन 3 के लिटरेरी, ड्रामेटिक, म्यूजिकल और आर्टिस्टिक वर्क, कोई कॉपीराइट के अंदर आते हैं ये सारे कुछ. जो भी हमने लिटरेरी वर्क किया है, ड्रामेटिक किया है, म्यूजिकल, वो पेटेंटेबल नहीं होगा, वो कॉपीराइट के अंदर आएगा. कोई किताब लिखी है, कोई डांस है, कोई गाना है. somebody asked me कि यूट्यूब पे कुछ भी गाना डांस वगैरह अपलोड कर देते हैं, देन कॉपीराइट का ऑप्शन आता है कि जो गाना गया है उसे लाइसेंस लेना पड़ेगा. यू हैव टू टेक द लाइसेंस फ्रॉम द सिंगर. किसी गाने पे आप या तो उसको म्यूट करके आप उसको सिर्फ डांस की परफॉर्मेंस को अपलोड करिए तो मिलेगा, अदरवाइज यू आर नॉट एबल टू गेट द कॉपीराइट फॉर दैट वन. आप अपलोड नहीं कर सकते. एंड सेक्शन 3I, 3J, मेयर स्कीम और रूल्स और मेथड और परफॉर्मिंग मेंटल एक्ट. कोई भी गेम खेलते हैं या कोई पजल सॉल्व करते हैं, वो भी पेटेंट नहीं ले सकते. एंड सेक्शन 3M, प्रेजेंटेशन ऑफ एनी इन्फॉर्मेशन इज नॉट पेटेंटेबल. लाइक हम यहाँ पे कोई प्रेजेंटेशन कर रहे हैं, उसका पेटेंट लेना चाहें तो हम नहीं ले सकते. 3M. एंड 3N, टोपोग्राफी और इंटीग्रेटेड सर्किट, दैट इज नॉट पेटेंटेबल. दैट विल बी फाइल्ड अंडर SSD LCC. एंड 3O, एनीथिंग रिलेटेड तो ठीक है, कोई ट्रेडिशनल नॉलेज है, मेडिसिनल प्रॉपर्टी जैसे हल्दी का मेडिसिनल यूज़ हम करते हैं तो उसका भी पेटेंट हम नहीं ले सकते, वो TKDL में आएगा. So these are 3A to 3O, around 11, है ना, 11, 12. These are the things which are not patentable. तो before filing or thinking for patent ना, we have to establish कि इसमें तो कोई है नहीं ना. Section 3 and 4 में हमको चेक करना पड़ेगा. That is checklist है ये. Invention आपकी नॉवेल हो, इन्वेंटिव हो, इंडस्ट्रियल एप्लीकेबल हो, और 3A से 3O तक में ना है. ये सारा कुछ आपको मिल जाएगा साइट पे, IP इंडिया की साइट पे भी क्या क्या है, क्या क्या नहीं है. ठीक है, यहाँ तक कोई ऐसा तो कुछ है नहीं इसमें, थ्योरी है सिंपल. सो अगेन in one shot, patentability criteria, novelty, inventive, and industrial applicability, and not fall under section 3 and 4. Then again, patent grant process. What are the process? सब कुछ हो गया, चेक कर लिया, पेटेंटेबल है, हमने इन्वेंशन बना ली, देख लिया सब ठीक है, एवरीथिंग इज ओके. देन फिर क्या करना है? पेटेंटेबल मैटर आपको मिल गया, ठीक है, आपने कुछ सोचा, यू हैव जस्ट थॉट एवरीथिंग इज ओके, सेक्शन 3, 4 में नहीं है, नॉवेल भी है, कहीं है नहीं, तो देन वी शुड थिंक फॉर द पेटेंट application, then we should apply for the patent. Then what are the process? We will discuss, but the first, it is a flow chart, like we will apply for the patent, then it will publish after 18 months. And if anybody has any objection, then he will raise the objection, and the pre-grant opposition is there. It is not written here, but that is in line. Then request for examination, we have to file within 48 months of the date of filing. These are all the statutory time limit by the Patent Act. 48 months के अंदर हमें फाइल करना ही है, RQ. Then examination report by the controller, FER is generated by the patent office. After that, and then after getting the report from the patent office, we have to respond to the patent office within 6 months from the date of issue of FER, first examination report. Then if controller satisfied, he will grant your case. Otherwise, he will call for the hearing. And after hearing, if he will satisfy, then he will grant. If any no opposition is there, and after grant, if everything is okay, if no post-grant opposition, then we will pay the maintenance fee. Up to the 20 years, you can pay the maintenance fee from date of filing. Please note that your case will be live from date of patent, 20 years from the date of patent, means date of filing. मान लीजिए आज फाइल किया है 1 फरवरी 2024, तो 20 साल कितना हुआ? 1 फरवरी 44, ठीक है. डेट ऑफ फाइलिंग से 20 साल रहेगा अगर हम मेंटेनेंस उसका प्रॉपर रिन्यूअल फी पे करते हैं. कोई ऑब्जेक्शन नहीं है. अगर ऑब्जेक्शन है तो यू हैव टू कंप्लाई द ऑब्जेक्शन रेज बाय द पर्सन थ्रू पेटेंट ऑफिस. कंट्रोलर आपको सारा कुछ देगा, आपको जवाब देना होगा. एंड ऑब्जेक्शन may be after grant and before grant also. Okay? There are systems, boards are there, 163 sections and 123 rules are there. For each and every step, there are the rules, there are the provisions, how to handle these things, which are the relevant forms. But in nut and shell, these are the gist. This is the process, this is the flow chart. ठीक है, यहाँ तक आप अप्लाई करोगे. ये सारे प्रोसेस होने में कितना टाइम लगता है? इफ लाइक वी अप्लाइड, देन 18 मंथ्स में आपका पब्लिकेशन हुआ, देन 48 मंथ्स में आपकी RQ फाइल हुई. तो नॉर्मल इफ़ यू आर ट्राइंग टू फाइल अ पेटेंट, ये 4 से 5 लग जाता है सर. ठीक है, नॉर्मल प्रोसेस. बट देयर आर सर्टेन रूल्स. हाँ, 4 से 5 सर, नॉर्मल प्रोसीजर. बट एक्सपेडाइटेड एग्जामिनेशन, देयर आर रूल्स वेयर यू कैन गेट द पेटेंट इन फास्ट ट्रैक मोड. 10 महीने में भी ग्रांट हो चुका है. So there are certain rules. Extra fee, yes. But right now there are rules for the SEM, small entity, startups and academic institutions and female inventors. 80% reduction in the patent fee. 80% आपकी छूट है. यानी कि आप अगर ONP में हैं तो 1600 रुपये में फाइलिंग हो जाएगी. कंप्लीट आप 1 लाख के अंदर ही पूरा 20 साल मेंटेन कर सकते हो. हाँ, मतलब these categories only. ठीक है. हाँ, if collaborating with others means industry, then not on a problem. He may be an inventor. The fees has been paid by the inventor, a female inventor. there's a deduction. But if you are assigning the rights, to the industry, then according to the industry rule, you have to pay more. Industry is which type? Startup, MSME, then you have to pay the nominal fee. But if it is not in this category, then you have to pay 8,000 rupees. So fees depends on the country, category wise. Okay? A lot of structures, things are there, but I'm just trying to give the overview. यहाँ तक ठीक है ना? Some important points, these are granted by the National Patent Office. Means these are the territorial rights. इंडिया में है तो इंडिया में रहेगा, यूएस का है तो यूएस में रहेगा. इंडिया में पेटेंट है तो हम यूएस में उसको बेच सकते हैं, ऐसी कोई इशू नहीं है. ठीक है? 20 साल के लिए होता है डेटा फाइलिंग से. राइट टू ओनर टू एक्सक्लूड अदर फ्रॉम यूजिंग, अगर किसी के पास पेटेंट का राइट है तो वो दूसरों को रोक सकता है. इफ़ यू हैव द पेटेंट for this particular pointer, then you can stop anybody to sell, purchase, या making or selling. आप उसको रोक सकते हो legally. Inventors obligations are there. And like we discussed, some forms are there. I'm just trying to, सब कुछ कर लिया, forms, yes. होता है, process होता है उसका, process. हाँ? नहीं, प्रोसेस 7 साल का नहीं होता. पहले था, अब सबका 20 साल हो गया है. 20 years फॉर यूनिफॉर्मली, इंडियन ही सभी जगह. फ्रॉम डेट ऑफ फाइलिंग 20 years, भूल जाइए. 7 साल, 15 साल पहले था. 2005 ऑनवर्ड्स इट इज़ यूनिफॉर्मली फॉर 20 years. कहीं भी पेटेंट की लाइफ है 20 साल, याद करिए. 7 साल, 15 साल पहले था, वो 2003 में आया अमेंडमेंट उसका. ठीक है, तो वो नहीं होता. 20 साल ही याद रखिए. Then after that, you have to, सब कुछ हो गया, आपने चेक कर लिया, then you have to apply the patent. So some forms are there, form 1, 2, 3, 4, 5. But remember these 1, 2, 3, and 5. These 4 forms are there to file an application in India. Okay? Form 1 for applicant, foreign patent, where inventors give is all details like name, address, everything is there. Form 1, you can see the templates in IP India sites. Form 2, that is main for the specification, where you have to write the specification. Form 2 में लिखेंगे सारा कुछ. Form 3, statement undertaking regarding the foreign applicant, कि आपने कहीं बाहर फाइल किया है उसके रिगार्डिंग एक फॉर्मेट है, उस पर फाइल करेंगे. Form 5, डिक्लेरेशन एज अ इन्वेंटरशिप. Form 5 में आप इन्वेंटर हो, इसके लिए एक डिक्लेरेशन देना पड़ता है. 1, 2, 3, 4 मिसिंग, 5. 1, 2, 3, 4, 5. 4 फॉर्म याद कर लीजिए. ये 4 को साथ आपको फाइल करना है इंडिया में, अगर फाइल करना है, प्रोविजनल और कंप्लीट. इसके बाद भी फॉर्म्स हैं. 18 रिक्वेस्ट फॉर एग्जामिनेशन के लिए है. 18A रिक्वेस्ट फॉर एक्सपेडाइट एग्जामिनेशन. देन पावर ऑफ अटॉर्नी फॉर ज्वाइंट केसेस एंड स्मॉल एंटिटी है, तो फॉर्म 28. सो 1, 2, 3 and 5, these 4 forms are required to file an patent in India. Either it may be provisional or complete. Provisional or complete क्या है? It's a type of application like if you are trying to make an specification, ठीक है, अगर आपको लगता है कि हमारा इन्वेंशन अभी कंप्लीट नहीं हुआ है, वी नीड सम डेटा एंड सम मोर टाइम, बट डिस्क्लोज हो गया है, या आई एम थिंकिंग टू गेट द प्रायोरिटी, तो देन वी हैव टू मेक एन प्रोविजनल एप्लीकेशन. प्रोविजनल में क्लेम नहीं होता, वी डोंट इंक्लूड द क्लेम, ओनली skeleton, whatever the prior, except then everything should be there. And after provisional, you have to file the complete within 12 months. 12 महीने के अंदर आपको फाइल करनी है. Yes. So, if you conceive any idea, then try to file a provisional application first to make the priority with the basic structure, like everything, abstract, whatever the abstract, title, everything, claim should be not there. After getting the data, you make the application complete and make the claims and file the complete one. ठीक है ना यहाँ तक? हाँ, within 12 months. So provisional application like that, and then complete specification have the title and everything with claims and all. Title, how we decide the title in publication? Na, title is very long, but in patent, the title is within 15 words. Quite indicative the subject matter, within 15 words, indicating the subject matter properly, and then free from any fancy words like novel, inventor's name. These are not included in the title. Like if we have invented a device, cooker, pressure cooker है ना, very simple, pressure cooker. then how can we make the title a pressure cooker for making rice? No, it should be very general. General means ki, a device cooking for grains. Grain is general. Broad होना चाहिए थोड़ा सा. So we should try to make the title very significant within 15 words. You can say ki much be broader, with free from novel, inventor names or any patent word. So title should be like that only. But it should be different from the publication. The publication mein kya, okay, we are using the long title and anything. Okay. So title is, we can write the title like that. Then these are the parts of the CS, जो हम patent लिखेंगे ना specification, उसके part है ये form 2 में. Then field of invention, from which field it belongs. Okay, this invention relates to whatever the area of biochemistry or whatever this. Okay, we start like that, field of invention. Then background and prior art. Whatever the prior art we had searched to establish the novelty, that should be included in this section, background and prior art. What are the drawbacks of that particular prior art? That should also be included, and how we are going to overcome. That should be included. And the closest prior art should be there. Closest prior art, समझ रहे हैं क्या होगा? Something very, some invention should be something very close to our invention. जो भी हमने किया उसके बहुत ही पास का कोई prior art हो, मतलब उससे related. That must be included in the background portion. Then summary. After that, we have to make the summary. These are the part of the specification only. Then detailed description, where we used, like we had made any device, like in pressure cooker banaya kuch bhi. Then how it is constructed, what are the parts, how it is connected, and how it is functioning. That should be detailed described in this one, detailed description. It's a very important. And if any working example is there, that should be there only. Example is very much essential for device cases. And in chemical cases, you have to make the structure and all, whatever. Examples and drawings should be there. And then drawings. For drawings, it should be on a 4-size sheet, systematically numbered, means for left-hand corner, your applicant name should be there, and right-hand corner, it is how much drawings are there, how much total number of pages, and it should be signed by the applicant, and it should be, no written material should be there in drawings. Rules are there, it should be black and white. So drawings like that only. ठीक है? ठीक है, यहाँ तो कोई problem नहीं है इससे यहाँ तक. Abstract is very important part of the CS. Complete specification या provisional का, this is an integral part. It should be within 150 words. 150 words. It should be what are the technical advancement you are claiming, that should be in abstract. How it is providing the solution of the existing problem, that should be in abstract. Accompanying with a figure. These all should be in concise summary. Along with the concise summary, it should be, you can comprising in abstract. Not like then publication. Publication in abstract ऐसे नहीं लिखा जाता. वहाँ figure एक से भी ज़्यादा हो जाते होंगे. But here it is in 150 words, clearly indicating the technical advancement, what are the technical problems you are going to solve, along with the figure. This is the beauty of this abstract, how to write the abstract. Then claims, this is the heart of the CS. पेटेंट का जो टेक्नो लीगल प्रोटेक्शन है हमको क्लेम से मिलता है. आपने क्या क्लेम में लिखा है ना, हाउ यू आर क्लेमिंग दैट इज द बेस्ड ऑन द क्लेम ओनली. जो भी आप क्लेम में लिखेंगे वो लीगल प्रोटेक्शन आपको वहीं से मिलेगा. एंड दिस इज नॉट एन वन डे एक्सरसाइज. क्लेम्स राइटिंग आर, यू कैन से इट इज अ प्रैक्टिस मैटर. Independent claim are there, dependent claims are there. Certain ways to write the claims. For device type of claim, we have to write different way. For chemical structures, we have write the claims in different way. So I am just giving the overview of the claims must be broader to narrower, and so many claims are good. and we have to write the product or process claims in one invention, and there should be only one invention's claim only, uniqueness should be there. So claim should be write in a very meticulous way, but how we write the claim, we can't discuss in one lecture, but that should be a separate, a very large, you can say scope is broader, that one. So some do's and don'ts are there, like working example along with data should be there. Your invention should be, novel inventive features should be highlighted. Abstract should be as per the limit of 150 words, and your drawing should be clear. Claims start from a fresh page, and these are some do's and don'ts are there. No descriptive matter in the drawings, we have already discussed. And here some more, you can say policy like thinking. This is on World IP Day in 26 April, on 26 April 2024. It is based on this theme, on SDG and IP. How we are thinking, how we are aligning this with the IP, and then we are aligning with the SDG goals. These are the SDG goals. 17 SDG goals are there. Right now, the new WIPO days on 26th of April, we have this vision like we have to align our IP with like these 17 SDG, like no poverty, zero hunger, good wealth balance. These are the SDGs, and we have to create, what we are thinking, we have to align, we are making the IP and how we are aligning this IP to these SDG goals. And this is the mapping of these SDG goals. 1, 3, 4, 5, 10, 16 are well-being, and infrastructure related to this one, and one health. This is all about this IP and aligning with the sustainable goals. This is the future pathway. So, like AI and ML we are discussing, we have no clear-cut guidelines for filing the patents for AI and ML. This is under means who is the owner of the AI, that invention, which are generated by the AI. It is under consideration right now. So this is all about something overview of patenting and all. So if, okay, it's okay. Anything, I'm trying to summarize all the things in a very nut and shell. Okay. Any question or any doubts? Because it is so. Yes, ma'am. नहीं हो सकता मैम, गन्स वो उसको आप वैसे नहीं होती, उसका दूसरा यू हैव टू शो दिस इज फॉर सेल्फ डिफेंस के लिए हम यूज़ करते हैं, तब होता है. यस यस यस, इंस्ट्रूमेंट में ही होता ना. हाँ. What? So let's say if you are a weapons designer, right, and you made some firearm or electrical applications for the government. हाँ हाँ. So can that be patented out under your name? नहीं, वो एक्चुअली में उसमें नहीं आएगा, वो सीक्रेसी डायरेक्शन में आता है. You have to take the approvals from the government and all, then it will a procedure for that one. You can't get a patent directly. अगर रेडिएशन वर्ड भी कहीं आपके CS में आ गया ना, रेडिएशन यूज फॉर दिस डिवाइस फॉर रेडिएशन, तो वो DAE से अप्रूव होके आएगा कि इट इज कम अंडर DAE एटॉमिक एनर्जी एक्ट एंड इट विल नॉट बी पेटेंटेबल. देन यू हैव टू रिमूव द रेडिएशन वर्ड एंड ऑल. सो देयर इज अ प्रोसीजर, सर्टेन प्रोसीजर फॉर दैट वन. Yes, yes. No. These are the territorial rights. Like you are using your, means if you file the patent in India, then you have the protection for India only. And if you import from the outside, then that is the property from outside. Pardon? No. That should be not. Yes, sir. Yes. Government government can use the patent for under, like if it is used for the public health, certain conditions are there, like compulsory अगर कोई epidemic है, for any natural disaster है, या कोई medicine ऐसी है जिसका use नहीं हो रहा है, तो उसको compulsory दे सकती है. 3-4 conditions उसकी हैं, एक epidemic में और कोई ऐसी वैक्सीन है या मेडिसिन है उसके लिए यूज़ कर सकती है, या गवर्नमेंट को लगता है कि वो कहीं पब्लिक हेल्थ के लिए, पब्लिक वेलफेयर के लिए यूज़ हो सकती है, देन ही कैन टेक इट. 100, 102 एंड 100 से 107 तक 7 सेक्शन है. देयर आर 7 सेक्शंस फॉर यूज फॉर द गवर्नमेंट. 7 सेक्शन है, एक नहीं है. 99, 100, 101, 102, 103, 104, 105, 106, 107. 8 सेक्शन पूरे. 8 सेक्शन में कई सारे एवरीथिंग इज शुड बी वेरी सिस्टमैटिक नंबर. गवर्नमेंट यूज़ के कितने सेक्शंस हैं, सीएल के कितने सेक्शंस हैं, सारे कुछ. ठीक है, कोई ज़्यादा उसमें तो ऐसा टेक्निकल तो है नहीं, बट सिंपल है. बट इफ यू फील दैट एनीथिंग प्रॉब्लमेटिक, यू कैन चेक इट. यस यस, हाँ, अगर आपका माइक यहाँ पे पेटेंटेड है ना, तो बाहर में इसका, लेकिन ये है कि बाहर अगर फाइल करता है तो ये प्रायर आर्ट में आ जाएगा ना. इंडिया में पेटेंटेड है, उसका प्रायर आर्ट वहाँ दिखेगा, तो वो सेम तो नहीं कर सकता, बस चेंज वेंज करके कर सकता है. इट्स अ ट्रू, होता भी है. देखिए, इट्स अ वेरी क्लियर. चेंज वेंज सेम तो प्रायर आर्ट में आ जाएगा, तो वो नहीं कर पाएगा, बस चेंज करके तो. हाँ, तो वो आपको दिखाना पड़ेगा कि एफिकेसी है उसकी, ये इसमें इन्वेंटिव स्टेप है, चेंज करके. तो यहाँ भी आप, इफ़ यू आर चेंजिंग दैट वन ना, उसमें आप क्राइटेरिया फुलफिल होने पड़ेगी तो हो जाएगा पेटेंट. इफ़ यू आर फुलफिलिंग द क्राइटेरिया, देन यू कैन फाइल अ पेटेंट, नॉट एन इशू. होता भी है. प्रोसेस में प्रॉब्लम यही तो है. प्रोसेस अगर आप लेते हो तो कोई भी थोड़ा सा चेंज करके, मान लो सोल्यूशन है, पीएच वैल्यू थोड़ा चेंज करके किसी ने प्रोसेस ले लिया, तो प्रोडक्ट और प्रोसेस दोनों लेना चाहिए. इट इज़ बेटर कि टेक द पेटेंट ऑफ प्रोडक्ट एंड प्रोसेस बोथ, बिकॉज प्रोसेस का उतना फ़ायदा नहीं है. प्रोसेस में कोई भी थोड़ा सा चेंज किया, रेशियो कुछ भी, और कंपोजिशन में रेशियो चेंज किया, देन ही कैन टेक द पेटेंट. ठीक है.
Now it's time to express our gratitude. We have a memento as a token of appreciation. I would like to request Dr. Nancy to kindly present a memento to our honored guest.
Thank you, Dr. Sushmita, for chairing the session. Now, we request Dr. Sunita to present a memento to Professor Sanjeev Thakur for, as a token of regard. Dr. Sunita to Thakur, sir. So with this lecture, this series comes to an end for today, but we are expecting more in the future. And nothing ends without acknowledging the efforts of the people behind the event. So now I request Professor Sanjeev Thakur to come forward and say a vote of thanks to everyone. Thank you, sir.
So thank you. I find all of you very delighted to listen to all the 4 lectures by the esteemed speakers, Professor Akhilesh Verma, Professor Bhavesh Dave, Professor Ram Singh, and Professor Gaurav Krishnan. I wished I could sit throughout the day and listen to all the 4 lectures, but because of having so many offices, I have to keep on shuttling around here and there. So I'm not that fortunate. I don't have the luxury of sitting throughout the day at one place and listen to all the fruitful lectures of all the honorable speakers. So the formality to give the vote of thanks, but really happy to be here. Organic Chemistry Series 2024 focused on the theme Harnessing Catalysis for Green Organic Synthesis. Very burning and interesting topic. This journey has been nothing short of exhilarating, and I'm extremely thankful to the collective efforts that have brought us to this moment. In my welcome address also, I had already mentioned that the effort has been tremendous, and they all deserve gratitude and congratulations, all the organizers. I'm also expressing my deepest gratitude to all the distinguished speakers. All of them are very busy persons. They are eminent scholars in their own fields. All of them belong to different areas, like Professor Verma. You have already gone through his talk, and Professor Ram Singh from DTU, and Dr. Dave, he's from pharmaceuticals, and Gaurav Krishnan, I think we need some more lectures from him, Dr. Gaurav Krishnan. So it has to be at the level of university, isn't it? Not just confined to the Department of Chemistry, but all of us need to learn from him how to go about patenting. I would like to express my sincere appreciation to the NSUT organizing team as well, without whose efforts this series would not have been possible. Our Honorable Vice Chancellor, Professor Srivastav, he is the motivating force and ready to give a green signal any time for these academic efforts. All the attendees, our esteemed colleagues, research students, master's students, many of the BTech students were also present in the morning. Your presence has added immense value to this series, making it a huge success, making it a vibrant platform for the exchange of ideas and knowledge. Thank you once again to everybody who has been a part of this incredible journey. Last but not the least, all those who are technical people who are involved in photography and doing YouTube business. I also thank all of them. And I look forward to seeing the positive impact our collective efforts will have on the world of organic chemistry. Thank you.
Thank you, sir. Thank you so much. Now this session ends with the tea outside. We have tea available outside. All right. Hello, ma'am. Hello, ma'am. मेरे वाले बिजनेस में तो नहीं थी। हेलो। मेरी बहुत दिल अनाउंसमेंट हो गई। अंदर मैम हाँ हाँ ले लो। इसमें मेरा एक ईमेल है, मैं उसे साइन आउट कर दूँगी।